Well, good afternoon again, everyone. Welcome back. This is the official start of the 2023 Astronaut Scholar Technical Conference. If you did not get a program, we'll bring some in. Anybody need a program? Raise your hand. I can bring some in. Lisa? Okay. We just a couple for the back. Okay, so we're really excited to get this kicked off, and I know you all have worked very hard on your presentations that are presenting today. Just a couple housekeeping items. It's a long afternoon, so we'll probably take a little stretch break at the discretion of our moderators, who I'll introduce momentarily. Restrooms, you've all probably figured out where they're at, down the hall, on the left as you go down the hallway. I would ask that please be cautious. We want to make sure you have really good audio, so that's why we have so many speakers set up, but be careful of the basses. So I know I've already stepped over this one numerous times because I was too lazy to go around, but don't be lazy like me. So just be safe as you're walking around and keep an eye on the bases of those speakers. I don't want anybody tripping on my watch. So without further ado, I want to introduce our moderators for today. I'm very excited to welcome back Carol Fitzgerald-Tyler. Carol is global leader of Wipro's ERP Global Change Management and Customer Success. She is also an AS trustee and an ASF mentor. So we are very excited to have her back. She does this every year for us. So thank you for being here, Carol. And her trusty sidekick is also returning, Ashley Hayden, Astronaut Scholar from 2018, Texas A&M. She is fifth year PhD candidate in neuroscience at Baylor College of Medicine in Houston, Texas. And Ashley is a gem of a scholar. She is a perfect example of how you stay engaged as a scholar, which you'll all get to do in a couple years. So thank you so much, and I'll turn it over to you.
Hi, everybody. Wake up. If you think this is going to be quiet, you're sorely mistaken. This is so exciting. This conference is so exciting. And for me, again, my name's Carol Fitzgerald-Tyler. I am delighted to be here. I am delighted to be part of the professional development part of ASF. I've given some of you, have attended some of my classes, quote unquote, master classes, whatever you want to call them. But the best part has been that those who wanted to follow up, we've been coaching together, we've been spending time together, we've been practicing together. And Stephen is like a perfect example. Stephen called me from a conference in Chicago, and here we are practicing together. Your material, so my parents are 85 and a half years old. Almost 86 years old. And I was telling my mother the other day on my way back from getting my hair freshened for this event, I said, Mom, one of our scholars is cutting DNA and inserting gap R. She says, isn't those the jeans you used to wear? No, Mom, not the jeans I used to wear. And I said, another one is creating reasons to reduce the amount of carbon outputs when you make ibuprofen. She says, you're allergic to ibuprofen. Thanks, Mom. Appreciate that. You're missing my point. But I believe my parents are actually dialed in somewhere in the remote link. So hi, Mom and Dad. So without further ado, though, all of you, in my humble opinion, are changing the world. You're not going to change the world. You are changing the world already. We are so lucky to have you, and we're so excited for your presentations. So thank you for being here. Thank you for taking the time to make them so wonderful. And a couple of teeny tiny housekeeping things, and Ashley will keep you on track. 5 minutes, most 6. We will cut off because we have 26 people to get through today. So be concise. Tell us your stories. We cannot wait to hear them. So thank you all.
Okay, couple other little housekeeping logistical things. So there is a clicker up here for those that'll be presenting today. There's a forward arrow that's pointing to the right. That's the one you use to go forward. And there's also a laser pointer, second button from the top. Okay, if you forget, just ask. I'll be right here. I want to take a quick note to say that you are all so brave for presenting. This is supposed to be very low stakes. This is you being surrounded by your peers and people who are going to think your research is really cool. And you're also the expert, okay? So I know a lot of you are worried about the questions you're going to get asked. I think you will all be very pleasantly surprised because you are an expert and you are here because you deserve to be here. So shake off the imposter syndrome, okay? I didn't even present my year because my imposter syndrome was so bad, okay? So be confident, come up here, take a deep breath. As we start to introduce your name, just go ahead and walk on up. And after we introduce you, we'll let you say your title and go forward, okay? Are y'all ready? Yeah, there we go. Get the energy going.
Thank you so much. And for our first 3 presentations, they will be remote. So the first one that we want to get started with is Madeline Blake, who I had the opportunity to meet in a very, very cold day in Minnesota in January. So Madeline, are you with us?
Yep, I'm here.
There she is. So please welcome Madeline Blake from the University of Minnesota.
All right. Am I good to start, guys?
Yes.
Okay, cool. I can't see the screen that's being projected, so I want to double check. All right. So, hi, my name is Madeline, and today I'm going to be discussing the importance of branching out, in which we showed that disrupted branched-chain amino acid metabolism exacerbates pulmonary arterial hypertension. I have no disclosures. So I'm going to begin this presentation by talking about the importance of right ventricular function in cardiovascular disease. RV failure is a risk factor for death in multiple cardiovascular diseases, including heart failure, cardiomyopathies, and the disease we principally study in our laboratory, pulmonary arterial hypertension, known as PAH, which is a condition that consists of high blood pressure in the lungs that ultimately results in right-sided heart failure and death. And currently, the treatment options available for RV failure are incredibly poor. So there's numerous effective therapies for left-sided heart failure and the left ventricle, including beta blockers, ACE inhibitors, and SGLT2 inhibitors. And at present, there are no available or effective therapies for the right ventricle and right-sided heart failure. So there's a dire need for innovation in the field, which is why we are doing this research. So we end up looking at branched-chain amino acids, which some of you might have seen before in a packaging like this. Very fun, watermelon flavor, seems pretty cool, right? Well, branched-chain amino acids are a type of essential amino acid that you can't produce yourself. You have to consume them through external sources like diet. And they gained our attention because recently several epidemiological studies have shown that elevated levels of branched-chain amino acid in your blood predict worse outcomes for multiple cardiovascular diseases, including heart failure, diabetes, and atrial fibrillation. So we thought, what if it's happening in our disease, pulmonary arterial hypertension, and could a drug that was aimed at reducing levels of these amino acids in the blood of our patients improve outcomes and provide an accessible treatment option? And so our study looked like this, where we had a 25-day translational rodent model that was utilizing a compound known as monocrotaline that when you inject it on day 1 of our experimental period, the rats will develop pulmonary arterial hypertension and associated right-sided heart failure over a 30-day course. And we allowed our animals to develop their disease for 14 days. And then on day 15, we then gave them our drug, BT2, which is a small molecule inducer of BCA catabolism. So essentially what we were doing is this drug was going to cause them to decrease the levels of BCAs in their blood by using them in metabolism. And the reason we had a translational rodent model for this study was because we don't see patients at the beginning of their disease. We see them somewhere near the middle, and we wanted to make sure our experimental studies in rodents reflected that. So our primary endpoints involved hemodynamic measurement of pH severity, echocardiographic assessment of RV function, physiological measurement of maximal exercise capacity, and proteomic, metabolomic, and lipidomic evaluation of lung tissue. And so on those first 2 endpoints, looking at how their lung disease changed and their right-sided heart disease changed, we found that BT2 significantly combated their pulmonary vascular disease and also their right ventricular dysfunction hypertrophy. So their heart disease and their lung disease got better with treatment. Then we wanted to see how their exercise capacity changed, because that's a major measurement we look at in patients in the hospital, and it's one that's very difficult to replicate in the lab. So we wanted to make sure we covered that base as well. And we found that BT2 treatment augmented their VO2 max, so how much oxygen they were able to consume during exercise, and improved their peak exercise capacity. And if you look over towards the right side of this graph, you can see that our animals not only went longer on the treadmill at higher intensity, but they also did it at a level comparable to our control animals. So that was a pretty amazing result from this study. They got a lot better. Then we actually looked at metabolism. We found that when looking at the proteins in the lung, we saw that BT2 reprogrammed lung mitochondrial protein regulation and multiple metabolic pathways associated with pH severity. So it improved some of the metabolic dysfunction that was happening in their lungs, and that improved their disease. And we also found that BT2 mitigated derangements in lung lipid metabolism. And that's traditionally what we see with this disease, where over time, as their lungs get sicker, they can't use their preferred source of energy, lipids or fats. And it looks like BT2 created a nice intermediate for that. So it rescued a lot of those deficits in lung lipid metabolism. So the lungs were able to have more fuel to at least maintain their condition. And so with this in mind, kind of a summary of what I've talked about is that our BT2 intervention, MCT-induced PAH model, decreased their pulmonary vascular disease severity, increased their RV function, increased their exercise performance on a treadmill, and increased pulmonary lipid metabolism. And so if I had one takeaway from this presentation for you all, it's that if you see a product like this in the store, maybe reconsider increasing your branched-chain amino acids before jumping right into this as a regular supplement. So thank you. And I'd like to acknowledge my lab, which has been very fundamental in this project, and my mentor, Dr. Kurt Prince, who has been amazing. So I'm now open to take questions.
Hey, Madeline, great talk. I was just wondering if y'all know what interaction is being had with the BT2, what protein interactions might be occurring?
Yeah, so that's a great question. So the primary mechanism for why we think BT2 works and what it's actually doing, because again, as I mentioned, it's a small molecule inducer of BCA catabolism. So it causes those branched-chain amino acids, rather than sitting in your blood, to be used in systemic metabolism. What it does as well, in addition to just reducing those BCAs sitting in the blood, is it actually suppresses mTOR indirectly, because these branched-chain amino acids have been shown to kind of exacerbate this pathway called mTOR that's a main component of the metabolic derangements and general pathophysiology of several diseases. PH is one of them, but also other diseases that are like cancer. And so it's suppressing that pathway as well. And we think that's a primary means by which these animals actually got better, and BT2 improved their lung disease and their heart disease.
Thank you.
Any other questions for Madeline? Madeline, thank you so much. We would love to be in person, but it's wonderful to see you. Thank you.
Wonderful to see you as well, Carol. Thank you all so much.
Our next presenter is also remote. Please meet Tara Young from the University of Washington.
Hi, everyone.
Hi, Tara.
Thank you so much for attending my presentation today. I'm here to talk a little bit more about DNA replication proteins, and a new protein that we discovered may play a role in a well-understood process. You've probably all seen how quickly bacteria can grow over old food in your fridge, or how fast cancer cells can metastasize. And this all has to do with DNA replication and how quickly it can occur. You may be familiar with the DNA double helix and popular representation in movies, and today I'm going to take you a bit deeper. But first, what does DNA look like in the cell? DNA in the cell is quite expanded in this image I'm showing you. And if you were to stretch the DNA in each of your cells from end to end, you would be reaching out over 6 feet. So how is all of this DNA able to fit inside of our cells? By a large level of winding and compaction. This is an image of all of the DNA stored in just one human cell. So how does DNA replication occur in this context? You can think of DNA as the earbuds that mysteriously get tangled in your pocket. If you were to pull further on each strand, it pushes a knot further down and prevents you from pulling them apart. Because DNA strands are wound in a helix and must be separated to be replicated, this leads to the formation of over-twisted and what we call positively supercoiled DNA ahead of the replication fork that looks a little bit like this, where the replosome, or the machinery that is used to replicate the DNA, is stalled by a knot in front of it called a positive supercoil, but all is not lost. Fortunately, our DNA is a little bit luckier than we are, and they have molecular scissors of enzymes called topoisomerases that cut and unwind this DNA. These enzymes are able to cut DNA at the region of the knot and relax DNA back into an easy to separate state, and then they glue the strands of DNA back together. Quickly growing cells like bacteria and cancer need high levels of topoisomerases to resolve the knots of DNA supercoils ahead of the replication fork, which makes them an excellent drug target because if you remove them, the cells will then die. But how do topoisomerases find their target DNA? We found that GAPR is a DNA binding protein that is essential to the model organism we study. GAPR binds specifically to this knotted, positively supercoiled DNA, and this made us wonder, does GAPR explain how topoisomerases find their target DNA? We found that GAPR does in fact stimulate topoisomerase activity. We identified this by adding tightly supercoiled DNA to a test tube and then adding topoisomerase by itself in the top gel and topoisomerase with GAPR in the bottom condition, and then measuring at specific time points how much of the DNA in the tube is still supercoiled. Because the supercoiled DNA, as I told you, is very compacted, it will run a lot further in the gel. So it represents that bottom band you're seeing. And towards the top is this relaxed DNA that's whole and circular. Over time, we only saw the disappearance of the supercoiled DNA when we added GAPR, which suggests that GAPR is necessary to the stimulation of removing supercoils. But how does GAPR interact with topoisomerases? I use the nifty interaction assay called the bacterial-2 hybrid, where if two proteins interact, they bring together pieces of a reporter that turn a colony red. I identified that GAPR directly interacts with both of the homologous subunits of bacterial topoisomerases. However, when I remove the last 13 amino acids of GAPR, it no longer interacts, which suggests a domain of interaction on these last 13 amino acids. My current work is trying to find how this interaction is occurring. I believe that there may be a salt bridge residue interaction within the last 13 amino acids, and that's my current investigation. Other projects in the GWO lab are looking to see if a similar system may also occur in ourselves and viruses and other organisms. By studying GAPR, which is the very first identified topoisomerase recruiter, my research holds tremendous therapeutic potential because topoisomerase inhibitors today are very important chemotherapeutic and antibiotic drugs. My research is revealing a novel pathway to inhibit this mechanism and expanding our understanding of DNA replication. This research will change how we are able to treat quickly replicating cells like cancer. Thank you all so much for listening to my presentation today, and special thanks to the other members of the GWO lab and collaborating labs at the Stowers Institute for Medical Research. This work is funded by the NIH, the UW Mary Gates Endowment, and the Undergraduate Research Program, as well as the Astronaut Scholarship Foundation, sponsored by John and Carol Tyler. I'm open for questions now.
Hi, thank you for presenting today. At the beginning of your presentation, you mentioned that if we took all of our DNA and unwind it, it would stretch about 6 feet. How does that change with age?
Yes, that's a really good question. So I think you're specifically talking a little bit about the ends of our DNA. In human cells, we have something called telomeres, which are rich in what we call the G and C bases of DNA. These bases are very good at bonding with each other, and they're much stronger. And so we have a cap on each side of our DNA called telomeres that will shorten in each round of DNA replication as each end of these caps is replicated. We don't grab all of it. So the DNA will shorten over time, but I don't believe in a significant amount because the telomeres do not make up most of our DNA. So I would hypothesize that it would be roughly the same length, give or take the telomeres over time. Yeah.
Thank you.
Of course.
Hi, great presentation. Do you look at all into how to localize GAPR to cancer cells that you would want to kill?
Yes, so right now we're still in the model organism stage of seeing what we can use GAP-R for. Kind of expanding on what you're talking about, we've looked to see if we can use GAP-R as a probe for positive supercoils to see where they're occurring in the cell. So there's definitely future potential to use that in cancer cells to see if this hypothesis of positive supercoiling preventing replication is the mechanism for top-2 inhibitors. So right now we're developing what we call GAP-R-GFP, where we have a fluorescent protein added to GAP-R and see if that will illuminate essentially where these supercoils are in a cell. So that could probably be applied, but that's, again, another future direction.
Tara, thank you so much. We miss having you in person, but your presentation was fascinating. Thank you so much.
Thank you.
Next, we'll again welcome a remote presenter, Will Jarvis from the University of Wisconsin. Welcome, Will.
Hello, it is good to be here. I am very sad that I cannot join you in person, but thank you very much for having me again. It's great to be back with the ASF for a second year, and it's truly an honor to be here. My name is Will Jarvis from the University of Madison. This research is underway at the University of Hawaii, and the initial RU funding was provided by NSF Grant 2050710. You probably won't remember that. I didn't remember it, but thank you to the NSF. I began this project at an RU in 2021, and I'm grateful for Dave to continue providing funding for me to work on it this summer. So let us begin. Galaxies are collections of dust, gas, stars, and dark matter, thousands to over 100,000 light years wide. They can have many different morphologies depending on the age and environment of the galaxy. We see disks like the beautiful M81. We see irregular emerging galaxies such as NGC 4438 or spheroids in the long, slow crawl towards death like M86 or NGC 3077. Understanding how galaxies come to these morphologies is an exciting and an active field of study. And I get to study black holes, one of the coolest things in astronomy. I focus on feeding supermassive black holes near the centers of galaxies called active galactic nuclei, or AGN. Material swirls around them, heated to temperatures of up to 1,000,000 degrees Kelvin, radiating energy shown by the yellow arrows that should hopefully spread out from the diagram. And this energy radiates out across the electromagnetic spectrum, creating characteristic emission features that we are able to see. These features in different colors based on how far they are away from the black hole can provide insight into the 3D environment of the black hole, helping us understand where it lives and how it feeds. The diagram at right shows the area around the AGN, while it is actually that emission from the accretion disk around the black hole. We know that AGN are connected to their host galaxies through mass and other relations, but it's still a matter of debate as to whether AGN emission and activity is directly related to the morphology. My work attempts to discover whether such a connection exists. So, which changed first, the black hole or the galaxy? To understand this, I had to ask a few questions. First, how do you get material for the black hole to feed on? Does the galaxy or the black hole evolve first? What role do galaxy mergers play in black hole evolution? If we know what the black hole and the galaxy are doing individually, we may be able to look for factors that link them together. Because morphology changes with cosmic time, it may be useful as a rough estimate of where in their lifespans a galaxy and its supermassive black hole are less than half the size of the full moon, but have been studied for, actually, can you go back a slide? Have been studied for hundreds of thousands of hours by telescopes, including the Hubble, Chandra, Spitzer, and James Webb Space Telescopes. Almost every single object in the image of Good South on screen is a galaxy, stretching 13 billion years into cosmic history. The telescopes provide beautiful data across the electromagnetic spectrum. This data allows us to study the multi-wavelength emission around the galaxy and from the AGN, while also providing high-resolution imagery we can use to classify the galaxies. For my work, I begin by plotting the combined emission of the AGN and the galaxy as shown at left. Each dot on that figure is another data point from a different filter, camera, or telescope. The x-axis of this plot is wavelength, with ultraviolet, very blue light on the left, and infrared or red light on the right. The shape of the plot can tell us a lot about the environment around the AGN and how it is feeding. After I've plotted the emission, I plot two images of the galaxies themselves from two different filters that allow me to see different features of stars and gas that would only show up in one or the other filter. This helps my team of undergraduates and I make precise morphological classification. Once both steps are done, we can analyze trends and attempt to do some statistics. So here's what we're left with. The left figure shows the emissions from all 196 AGN-containing galaxies in our samples, colored by their intrinsic X-ray luminosity. Basically, the bluer it is, the dimmer it is. The black hole is either more obscured or just fainter, while the yellow stuff is really bright, some of the brightest objects in the universe, in fact. We see many different shapes present, from the kind of U shape to relatively flat emission trends. Ultimately, we sort these based on the shape of the emission to tell us about different groups of AGN. On the right, we see a basic breakdown of the morphologies. Most are classified as disks or spheroids, but there are a solid plurality of irregular galaxies as well. And those are just galaxies that show lots of disruption. Ultimately, we can split these into different panels to study different populations of the galaxies. And what we see is that there are some trends that seem to imply that as the emission gets brighter, you get more spheroidal objects. Ultimately, this kind of agrees with our theories that you'll get bright black holes after mergers and near the ends of the lifespans of some of these galaxies, while the fainter ones are more obscured and still in kind of disk galaxies. For more information, I will direct you to a paper that I'm hoping to publish later this year, and I'll be sure to post it in the alumni Facebook page. Thank you very much.
Thank you, Will. Who has a question for Will? There's one in the way back.
Hi, Will. Incredible research. I actually personally work with quasi-periodic oscillations and active galactic nuclei, and I love hearing the word AGN put up in other research. So I'm curious to hear if you also have encountered any or look at energy fluctuations in your morphological classifications. I know that when you're looking at morphology, you don't look at the long-term changes like that, but have you actually encountered any of your objects that do have those fluctuations that are whether regular or quasi-periodic?
Yeah, so we don't really dig too much into the periodicity of these sources because GOODS has been observed for so long. In this case, Hubble has looked at these for something like on the order of 50,000 to 100,000 hours. So any periodicity is going to get smoothed out over time in the photometry. So we don't really look at that. All of our photometry that we're basing this on is going to be summed together over years of over years of observations. So we don't really dive into that too much.
I was actually,
good question.
But I do want to add, we do see, I use RXT research, and we do see some periodicities that occur over years. So that's why I was kind of curious as to if you saw any of those longer terms through the observations, longer term periodicities.
Oh yeah, I mean, we haven't delved into that. I think that would have to be a different project to really dig into it, or it could be a nice follow-up piece to look kind of galaxy by galaxy. Right now, we're just interested in getting kind of the larger scale, large as 196 galaxies, but getting the larger scale statistics done.
Thank you very much.
Thank you, Will, so much. We wish you were here with us, and thank you for a very insightful presentation. So our next presenter will be in person. Her name is Katie Trainellis from North Carolina State University. But one small piece of housekeeping again for our presenters. We have Michael here in the front row. Michael is going to hold up the right finger. Joke, wake up, hello, is this thing on? He's going to hold up a finger when you're about 1 minute out from being done. So please keep an eye on Michael, and thank you, Michael, for doing that. And welcome to Katie.
Okay, can everyone hear me okay? Okay, awesome. So I'm Katie, and today I'm going to be talking to you about my work, which is on investigating the residue specificity of P300 in human histone peptides. So I'm going to wait for the slides to come up. Okay. So in our cells, our DNA is compacted into chromatin, and the basic subunit of chromatin is called a nucleosome. Now, the nucleosome is made up of an octamer of histone proteins that are wrapped in DNA, and the ends of these histone proteins are flexible, and they extend off of the nucleosome. They're called N-terminal tails. And on these tails, there's a number of different reversible chemical modifications that can be made. So they can be acetylated or methylated, and those modifications, which are called epigenetic modifications, are very important because they change the structure of chromatin, so that impacts gene expression in our cells. And when they're not regulated properly, they can contribute to a number of different disease states including cancers and neurodegenerative disorders. So the enzymes that add these modifications onto histone tails are called epigenomic writers. And my project is focused on P300, so that adds acetyl groups onto lysine residues on these tails. So why do we care about characterizing writers like P300? Well, they've actually been used in gene editing complexes, so think DCAS9 and CRISPR, and those have therapeutic value. So our goal is to be able to understand the editing capability of P300 in order to design better epigenomic editors. And we want to do that in a high throughput manner. So that leads us to the research question, which is, how do the amino acids around a lysine residue impact that lysine's acetylation by P300? So to study this, I screened a library of 7-mer peptides based on acetylation levels. So basically the sequence, it's 7 amino acids long. The center amino acid is a lysine, so that's what that K in the sequence is, and then the 3 ends on either side of it represent randomized amino acids. So that gives us a theoretical sequence diversity of 6.4 times 10 to the 7th different potential protein sequences that we're looking at. So how we screen them is through yeast surface display, and how that works is we have a yeast cell, and on the surface of that yeast cell, one of those 7-mer peptides is immobilized. And it's a library, so in the culture, there's a bunch of different yeast cells, and each one has its own unique peptide on the surface. But again, only some of those peptides are acetylated, and our goal is to isolate the sequences that are. So how we do that is we take our cells expressing our protein, we label them with antibodies, then we sort them based on acetylation levels using fluorescence-activated cell sorting. We collect a positive and a negative population, then we use, the pointer wasn't working, flow cytometry to make sure that sort was successful. Then if it was, we extract the DNA, send it for sequencing, and then do some data analysis. So in these plots I'm going to show you, acetylation is on the x-axis, and anything in the lower right-hand quadrant, which will be colored in, is positive for acetylation. So before I sorted, I just wanted to look at the library in general and see how much acetylation was happening, and it was about 8%. So with that information, I went ahead and sorted, and if you compare these two plots, you can see that the one on the right has a lot more cells in that acetyl-positive region, which is good. That means that we did enrich for acetyl-positive cells. It went from about 8% in the middle plot to 45% in the right. So then as a control, I ran the negatives, and we didn't see any acetylation happening there, so that was really good. So we extracted the DNA, and we sent it for sequencing. So when I got those sequences back, I cleaned them up and generated these logos plots. So how a logos plot works is it displays the letter corresponding to an amino acid at a particular residue, and the height of the letter corresponds to its frequency in the population. So the bigger the letter, the more often it's occurring. So in the negatives, we saw a lysine where it was supposed to be, but we didn't see anything else showing up, which makes sense because over 90% of this library was not acetylated, so we would expect the negatives to be randomized. But when we looked at the positives, we did see that there were amino acids that were enriched, particularly at these residues directly before and after the lysine, so 3 and 5 respectively. So we wanted to zoom in and look at those in particular. So we looked at residue 3, which is directly before the lysine, and what we did was we looked at the frequencies of the amino acids there, and then we normalized them by random probability, and what we found was that glycine was highly enriched. So glycine is the smallest amino acid. That's the chemical structure up there on the screen. Then we did the same analysis at residue 5, and what we found was that tyrosine, tryptophan, and phenylalanine were all highly enriched, which is interesting because they're bigger and they're also the 3 aromatic amino acids. So moving forward, I've sorted this library again with a different panacetyl antibody, and I'm in the process of comparing those data sets. And I'd also like to do this workflow with the click chemistry reagent, which eliminates the need for antibodies. And kind of the big picture takeaway is that we've created a high throughput system for studying epigenomic writer specificity, and this can be adapted to different proteins, different writers, different modifications, provided that you have a method for screening for that modification. And this system has allowed us to identify that there does appear to be some amino acid preferences for acetylation by P300, and that has potential application in designing better epigenomic editors, hopefully for therapeutic purposes. With that, I would like to thank my labs, which is the Kyung Lab and the Rao Lab, and also my graduate mentor, Allison Waldman. And if anyone has any questions, I'm happy to take them.
Hi, great talk. I'm wondering if you have any theories as to why there's enrichment of glycine on one side and these aromatic amino acids on the other, and just kind of what that means for P300 and its localization.
Yeah, so great question. I think that it probably has to do with the active site of the enzyme, which we're not doing any structural studies, so there's no way for us to actually look at that with this project. But glycine is also, like I said, the smallest amino acid. So one idea I had is that maybe that's potentially leaving that lysine residue free for whatever's happening in the active site, and it's just easier for P300 to get in there and add that acetyl group on. And as for the aromatics, potentially there's like a hydrophobic pocket in the enzyme. So those were my theories on why those were the amino acids that were being enriched.
Do you know the prevalence of the glycine-lysine in your target protein?
Okay, yeah, that's a great question. So we are focused on kind of the big idea for doing this was the interest in the histone N-terminal tails and studying that. So since we're still kind of sorting and gathering data, we want to do this with a couple different antibodies to make sure that the antibodies themselves are not causing these sequence preferences we're seeing. But that is a plan that we have. We're going to go through and look at all of the known sites and histone proteins that are acetylated and see do those sequences match what we're seeing. And we would also like to do that with the whole human proteome and just kind of see what other proteins match these sequences as well, because P300 acetylates more than just histone proteins.
Really interesting. I was curious if you could speculate how this could improve things like CRISPR activation. Do you think that it would greatly improve the efficiency and you could maybe use less, or how do you see this being implemented in something like that?
Okay, yeah, that's a really good question too. So I myself do not actually work with CRISPR. The idea for how this would be implemented is it would kind of like be tethered to CRISPR, and CRISPR would be deactivated so it wouldn't be cutting the DNA, but you'd have the binding recognition, and you would add P300 and potentially add an acetyl group back into an area where it's like not present, because typically acetylation is associated with higher gene expression. So say you have a tumor suppressor gene in your cells and it's turned off when it needs to be turned on, and we could have CRISPR go in and deliver an acetyl group and turn that back on so your cells can potentially fight cancer better because your gene to do it is turned back on now.
That's amazing. Thank you.
Yeah.
Thank you so much, Katie. Very interesting. We appreciate it.
Thank you.
I'd like to welcome our next presenter, Stephen LeBaum. Stephen is going to give us a talk from his work at the University of Chicago. Welcome, Stephen.
All right, good afternoon, Hello, there we go. Hello, there we go. All right, good afternoon, everyone. My name is Stephen LeBaum. I attend the University of Chicago, and my passion is chemistry. Today, I would like to talk about a new, more efficient method of chemical synthesis. Did you know that every year we produce 45,000 tons of ibuprofen? Yet producing these 45,000 tons of ibuprofen produces waste, 150,000 tons of it. Think of what that does to our environment and the pockets of the people who have to pay to dispose of it. But what can we do about it? Today I'd like to give a two-step solution. Step one is catalysis, which already exists and is being used. Step two is something I'd like to call catalysis 2.0. This is new, and this is the focus of my talk today. So what is catalysis? Say you had this molecule, and you wanted to add 2 hydrogen atoms to it to turn it into this molecule. Normally, you could do that with a proportional amount of reagents, where a reagent is just something that converts one chemical into another chemical. But using a proportional amount of reagents generates a proportional amount of waste, which we quantify with something called atom economy. Atom economy basically just involves counting up all of the atoms in the reactants and seeing how many make it into the final product. In this case, it's all of the pink ones. That's 81%. The other 19% of the atoms are the gray ones, and those go to waste. But how can we get this even higher? Well, we use a catalyst. A catalyst is not only something that accelerates a reaction, but something that is not consumed in the process. This means that a catalyst can grab 2 molecules of reactant and turn it into your product, and then it can grab another 2 and another 2 over and over again. And as the amount of product produced grows to far outweigh the amount of catalyst still in solution, we can approach. We'll never quite get there, but we can approach 100% atom economy. So problem solved, right? Not quite. Most catalysts are prone to a number of deactivation pathways. Although they're ideally not consumed, that is just ideal. A common deactivation pathway is when 2 catalyst molecules bump into each other and convert into some kind of inactive byproduct. But what if we could hold the catalyst molecules in place so that they couldn't bump into each other? This is catalysis 2.0, and this is where connects come into the picture. So if you're not familiar with connects, think of Legos. Connects are just longer and skinnier Legos. We build a flat sheet of connects. We scale it down literally 1 billion times. We affix our catalysts to the junctions. Done. Now they can't bump into each other. Of course, they don't make connects that small, so the actual structure looks more like this. And it's called a metal-organic layer. Let's look at an example. Suppose we had these molecules and we wanted to convert them into these molecules. We can do that with this catalyst. However, this catalyst is not very good at its job, so to even get it to work, we have to mix in a light harvesting molecule that can convert solar energy into catalytic energy. We attach both of these molecules to a metal organic layer. This way the catalyst, all of the gold balls, can't bump into each other, so they're stabilized, and the catalyst and the photosensitizer, the green part, are held very close together, facilitating efficient energy transfer. For all of the organic chemists in the room, these are all of the reactions that you can run. If you're not an organic chemist, this basically means that this system is capable of running a large variety of reactions, potentially expanding the scope of industrial and pharmaceutical reactions that this research can be applied to. So where do we go from here? Well, I just walked you through a gold catalyst. The link down there is to a cobalt catalyst, but we'd like to see what other kinds of metal catalysts we can also integrate into metal organic layers. We'd also like to expand the number of types of geometries. Metal organic layers are a subclass of 3-dimensional metal organic frameworks. And whereas we have tens of thousands of metal organic frameworks, these are the only 2-dimensional single molecular layer thick metal organic layers that we have to date. So we'd like to see what other kinds we can make and what possibilities those might hold. If you remember anything from my presentation, it should be this. In every chemical reaction, some percentage of the starting materials becomes waste. Chemical companies reduce this percentage with catalysis. And chemical companies can make catalysis 200 plus times more efficient if they embrace metal organic layers. I'd like to thank my lab as well as the Astronaut Scholars Scholarship Foundation for giving me this opportunity to present. I'll still be working on this research for another year, so if you're interested in talking about me any further with it or about anything else, please feel free to reach out on LinkedIn and I'll take any questions. Thank you.
Hello, Stephen. Hello? Okay. I was wondering what the next best, in your opinion, or anyone's opinion, type of metal organic compounds would be developed? Like, what metal would be the best for it in your mind?
So it's purely just specific to the kind of reaction you're trying to run. So if I go back a couple slides. Here we use a gold catalyst because gold is what can make this reaction happen. So it doesn't really depend on the kind of metal. Any kind of metal catalyst can be integrated into a metal organic framework theoretically if it has a carboxylate group on it. So where we see the squiggly line at the bottom of the gold AU, there's a carboxylate down there, and that's what binds it to the framework.
How does the spacing of the framework affect efficiency, and what's the distribution of the length of the framework?
Right there. Okay. Sorry, could you repeat that one more time?
How does the spacing of the framework affect the efficiency of the catalysis, and what's the distribution of the length of the framework? I mean, do you have uniform spacing, or is there a distribution of the spacing?
Yeah, no, so we don't have uniform space, and that's definitely one of the issues. We have about 8% catalyst loading, and we have about 16% photosensitizer. That's still good enough to get 200 times increase in efficiency, but it would theoretically be much higher if we could get that number closer to 100. As to the spacing, I don't have the diagram here, but the catalyst and photosensitizer active site is actually about only 1 nanometer, which is very good for efficient energy transfer.
Any other questions? One more, very quick.
How easy is it to manufacture these layers? What's the current holdup in using these?
Yeah, that's exactly the current holdup in using them. So they are quite fidgety to manufacture. That's one of the reasons why we only have 2 so far. We understand a little bit of the way that they're formed, but not nearly enough to be able to predictably make them in different kinds. So scale is going to be the Achilles heel right now, but we're hoping that that can be solved with more research.
Thank you so much. Very interesting. All right. Our next presenter that we're welcoming is Brandon Tupea from the Virginia Polytechnic Institute and State University. Welcome, Brandon.
Hey everyone. So my research is on how atomistic simulations can predict performance characteristics of a ladder polymer known as canal Me2F for gas separations. So to start off, industrial separations simply consume a lot of energy. That's about 10 to 15% of the world's energy consumption. If we look at just the industrial sector, separations consume about 45 to 55% of that. If we look at just membrane technology, we can reduce that by 90% and also then save CO2 emissions and also save money, so it's a win-win. So what we'll be looking at here is canal polymers. So that's the canal polymer I'm looking at, canal methyl 2-fluorine. First off, we'll look at how canal polymers are currently pushing the limits of membrane technologies. So what current membranes are being used are down there. What we want to do is push past that line, which is called the Robeson upper bound. So after 1 day, canal polymers are right near that upper bound. After 150 days, they're actually past that upper bound, which, out of scope of this presentation, is actually really unique. But however, there have been no simulation studies that have been performed on any of these canal polymers, which can help with high temperatures, which are inaccessible to experimental, uncoupling sorption and diffusion coefficients, and also analyzing new directions for these canal polymers. For example, what types of new atoms can we bond to them? So the type of simulations that we largely run are known as Monte Carlo simulations. So there's stat-mech analysis, which is used from repeated random sampling. It's an attempt to solve deterministic problems with randomness. So what we use is grand canonical Monte Carlo with a partition function, which we can see here, where we're allowed to fluctuate the chemical potential, mu, the volume, and the temperature. We have an absorption universe, which is made up of the polymer and an infinite gas reservoir. We can do reservoir to system moves, which are known as insertion and deletion. And we can do within system moves, which are known as rotation and translation. All of this is governed by the Metropolis acceptance criteria. So we'll look at some material properties first. First off is pore size distribution. So the exact simulated geometric pore size distribution is in pink, while the experimental through non-local density functional theory is in black. First off, we see that the geometric is below the experimental, and that is expected because NLDFT is known to overestimate pore size. So while it shows porosity, it doesn't give a good example of the sieving that we're going to be looking at here. Second, we look at wax, or wide-angle X-ray scattering. So at that inverse length of 1, we see the significant peak that we would have seen with the experimental. However, at high length scales, so low Q, we actually see that it doesn't follow. That is understandable because these distances are simply too large for the simulation size that we're looking at of about 6,900 atoms. So let's look at some adsorption isotherms. So we'll start with the small, simple gases, which allow for error to be attributed simply only to the polymer system. And we see that for hydrogen, oxygen, nitrogen, and methane, we see really good results. However, when we look at these larger gases, for example, plasticizable gases like CO2, we actually see that we don't follow a qualitative fit at high pressures. So what we can't see is the plasticization effects. Plasticization effects are essentially how the polymer swells as more CO2 is loaded into it. So what we say is the polymer must be able to relax in the presence of adsorbent. So to do that, we iterate grand canonical Monte Carlo with molecular dynamics, which allows for time scales to be used and for the system to relax. And with that, we can see a better qualitative and quantitative fit when we use the flexible framework there. At high loadings, it still does over-predict. However, that is understandable because we have lower densities than experimental. So let's look at, in lieu of a traditional conclusion, we're going to look at some cases that this can be applied. So first off, we're going to look at multi-temperature absorption. So you'll see here that we don't have dots. We actually have lines, and that's because we're using the dual-mode sorption isotherm right there with the Koros binary gas approximation. So looking at CO2 and CH4 in the presence of each other, we can also then see that dotted line, which is the selectivity. But then we can look at, we switch to fugacity because we want to accurately model these intergas potentials here. When we look at 328 Kelvin, we actually see that the selectivity increases slightly at low fugacities while decreasing at higher fugacities. So this gives an understanding of what temperatures we want to run these adsorption isotherms at. Next, we'll look at C3 separation, so that's propane and propylene. We see that at 288, it's higher than 308 and 328. That makes sense because adsorption is an exothermic process. However, all the ideal sorption selectivities are largely around 1, which really means that any separation potential is due to diffusion without any adsorption help, is how we'll look at that there. With that, we'll do some acknowledgements. So my PI, Dr. Smith, my mentor, Jing Ying, the canals team over at Stanford, the DOE for funding, and then the entire Smith lab. And with that, we'll open it up for Q&A.
Thank you. We do have time for one question for Brandon. Does anybody have a question?
I actually can ask a question. So this is probably a little bit naive, but what is it about canals in particular that are sort of important for this work? Like why focusing on canals as opposed to another polymer group?
Yeah, so these canal polymers are 3D contorted. So what essentially happens here is that they have really rigid backbones where they can make these unique shapes, which allows for a lot of space between the polymer to allow diffusion to happen, while also, because of these shapes, allows for a lot of selectivity between gas sizes.
Okay, thank you.
Thank you, Brandon. Very appreciated. We'd like to welcome next. Am I on? Yeah. I'd like to welcome next Max Shepard from the Colorado School of Mines. Welcome, Max.
All right, hi everyone. So today I'm going to be talking to you guys about some renewable energy research that I am working on. Specifically, we're looking at water electrolyzers and trying to understand the interface and trying to explain some of the performance things that we are seeing in terms of some of the results that our partners found. So this is in collaboration with the Colorado School of Mines, which is where I attend, and it's also in collaboration with the National Renewable Energy Laboratory, which is known as NREL. And this work is being published in the next couple of months just from some of the findings that we found here. Cool, so before diving super deep into what my project specifically is, this is just a little bit of background that I think is really useful to know before we kind of dive into it. So first off, what is green hydrogen? So green hydrogen is a way to produce hydrogen using renewable energy. And then this process is usually conducted through electrolysis, which essentially is applying electricity, typically with that renewable energy if we're looking at green hydrogen, to create hydrogen and oxygen from water. So we're using electricity, separating the hydrogen and oxygen out of water. And then the polymer electrolyte membrane water electrolyzer is focused on capturing the hydrogen and oxygen in chambers. and then using that hydrogen that's captured for different applications such as hydrogen fuel cells, which is an energy device that its only output is H2O as an emission. So it is very great for our environment and for potentially cars to run on in the future. So a quick project overview. So I'm coming from a characterization standpoint. So NREL did a lot of testing to many different samples, and some of these tests include mechanic abrasion tests. So when I say mechanic abrasion tests, you can think of like sandpaper. We had different grits of sandpaper that were homogeneously or uniformly applied to the sample. And then we also had, chemical tests. And those chemical tests were basically just chemical processes like etching that were applied to the sample. And before we move on to some of the results here, NREL did a lot of these tests kind of from a perspective of, hey, we're going to destroy these samples and try to just observe how much the performance is going to decrease. And we actually saw the performance increase, which was really shocking, and why NREL came to my lab to further investigate and understand the chemistry of what's actually going on. So in that blue box, we can see all the different parameters of the mechanic abrasions. Once again, those are like your, think just sandpaper grits. They're all just kind of like different sandpaper grits. And the techniques that I use were SEM, which is scanning electron microscopy, which is awesome for kind of just understanding morphology on the surface of our samples. And then energy dispersive X-ray spectroscopy, which is known as EDS or EDX, which is used to study elemental composition on the surface as well. So here are just a few pictures of my research, and these are SEM, scanning electron microscopy. So think of just a really high-powered microscope under vacuum. And we can see that there's a lot of different variations in all these samples, and they're all being treated with different sanding grits. So we can see that there's some thinner abrasions, there's thicker abrasions, there's deeper abrasions, and all these different abrasions can be attributed to some of the performance results that we are seeing. So we were able to go to NREL and say, hey, this is why you are seeing what you're seeing. And we can't really go into the why here in the setting because it's so fast, but it's just really cool to kind of look at the pictures. And then this is kind of the characterization of the catalyst layer. So in your water electrolyzer device, you have many different layers. And this catalyst layer is pressed against something called the porous transport layer. So on that previous slide, we were looking at the porous transport layer, which is just another part of the water electrolyzer. And when they're pressed together, like they would be in in the actual device, then they're tested at high pressure, and we can see that there's some results that we saw in the energy dispersive X-ray spectroscopy, which is the color diagram. And then you can also see it in the scanning electron microscopy, where parts of the porous transport layer peeled off onto the parts of our catalyst-coated membrane, or our catalyst protective layer. And you can see that in that red there, where we have platinum. And platinum should not be there. It should just be iridium. So that's how we know that there was some degradation and some durability issues after the testing was conducted. Going down to the chemical treatment, we can see that there are kind of those pits. You can kind of see those holes, which weren't there initially. And that was really surprising to NREL when we reported this. And we believe that is caused by some of the chemical processes, which is etching, that were applied to the samples. And that can show us that there were more contact points between the two surfaces of our material, which can help us explain why we saw an increase in performance. But we can also predict that there's going to be a decrease in kind of the longevity of this water electrolyzer device. So these are just some of the conclusions that we are able to make from this research, and these are not all of them, but just some of the main ones. So first off, SEM and EDS, which is the two characterization techniques that I used, are a great tool for trying to understand the interface and explain some of the performance results. We can also see that mechanic treatments, this is kind of intuitive, have caused different types of abrasions on the surface with different sanding grit treatments. Thirdly, we can see there is a difference between the mechanical and the chemical treatments. So the chemical treatments had that etching and those like pits, and the mechanic treatment had the different levels of abrasion. And that's going to impact how the surface is interacting with itself and the performance as a result. We can see that some of the mechanic treated samples, of course, have different abrasion sizes. Once again, I already mentioned the pitting. And then lastly, that the PTLs, which is the porous transport layer, that was mechanically treated, had more peel off from the the mechanically treated parts had more peel off from from the protective layer than the chemically treated samples. And that's really helpful for NREL to know, just so that we can, like in the future, when we orient our research or try to bring this to a more commercial scale, we can better understand what routes we want to take. So we probably want to stay more towards the chemical side because there is more of those contact points, which translate to higher performance. And with that, I'll open up to questions.
Thank you, Max. Who has a question for Max? There we go.
Yes.
I guess I had a question. This is more general about like electrolysis, I guess, but my experience with these kinds of electrochemical cells where you separate out things is that water is not pure. So if you're separating the hydrogen and the oxygen, what do you do with the waste?
So that's a great point. And so from my perspective on this research, we're not coming from it from a standpoint of, is this the right process to do? We're more coming from a characterization standpoint. So we're trying just to serve as somebody who can help our customer better understand why they're seeing what they're seeing. So I don't really have a good answer for that, and it's kind of out of scope of this specific project here.
Okay, thanks.
Thank you so much, Max.
Awesome, thank you guys.
Next, we'd like to welcome Aiden Kemp from Auburn University. Welcome, Aiden.
Good afternoon, everyone. My name is Aiden Kemp. I'm a rising senior in aerospace engineering and biochemical engineering, and my research focus is specifically on waste management systems going into ECLS, which is Environmental Control and Life Support Systems Engineering, how we can go ahead and continue to recycle nutrients or supplies that we have in space and to make the most of the materials that we have there, to be able to recuperate as much energy as possible and to be able to continue to push forward for longer duration space missions. And so the research that I was conducting was in the production of biofuels from blends of waste plastics and biomass. And so in 2021, America produced 40 million tons of plastic, 34 million tons of which went into landfills. I'm sure most of you have heard about the Pacific plastic patch and all of those waste plastics there, and no one wants to kill sea turtles. And so what we should do is we should find a different way to be able to use these waste materials for something actually beneficial. We can turn them into rocket fuel. Kerosene, for example. For those of you who saw the Falcon 9 launch on Wednesday night, that uses kerosene, and we can produce kerosene from those waste plastics. And so the process we use for that is pyrolysis. And so my purpose of my investigation was to go ahead and see if we combined plastic waste, polystyrene, that is what you would use in a styrofoam cup or plastic utensils, a lot of things that are commonly used in disposable consumer products, and combined it then with longleaf pine that is incredibly prevalent in the South. It's the most common tree in Alabama, Mississippi, and Georgia. There's a lot of it. And so combine those two feedstocks together and go ahead and perform pyrolysis on this and produce fuel from it. And then with that fuel, those pyrolysis oils that I produced, then to go ahead and characterize those to determine the fractions that we can produce of diesel fuel or of kerosene or of other products that are very valuable for putting into chemical engineering processes. And so what pyrolysis is to heat a material at a very high temperature, at atmospheric pressure, and to go ahead and break down the polymer chains into the individual monomer components. And so I investigated changing the blends of the feedstocks. I used pure polystyrene, a blend of 50% polystyrene and 50% pine, and then 100% pine. I varied the temperature between 450, 500 degrees Celsius, and 550 degrees. And then I used catalysts, both no catalyst, red mud, which is a waste product produced from aluminum ore processing, and then ZSM5. And so I developed a pilot scale pyrolysis reactor just using stainless steel tubing and a small bench scale furnace. And the process for that is shown here. And so there's a nitrogen purge gas which goes and pushes the pyrolysis oil fumes into the condenser train, which are then condensed and collected. I also developed an ESP, an electrostatic precipitator, to go ahead and to pull any of those waste products out of the atmosphere. That way we're not putting that out into the environment. And so The results we were specifically looking for and the process we were trying to optimize was the production of oil, because that is what contains the energy. And so we noticed from the oil that higher temperatures tend to produce more oil, and then catalysts actually tend to decrease the amount of oil produced simply because they actually further gas cracking. And then here, this is the thing that I really want to point out. So from pure polystyrene, so your plastic forks, I just ground them up and put them into this, and the energy of that oil collected was close to 42 megajoules per kilogram. And just for reference, crude oil is between 40 and 47 megajoules per kilogram. Diesel fuel is between 42 and 46 megajoules per kilogram. So a plastic fork was able to produce as much energy as diesel fuel. And so the applications for this, the applications for this is that we can go ahead and take waste materials, and instead of putting them into landfills, putting them into the ocean, disposing of them, losing all of that value that is stored within them, we can actually produce energy from this. We can go ahead and apply this into space. The ISS has a lot of plastic packaging that they just go ahead and put into the Cygnus capsule, and it burns up on reentry into the atmosphere. Whereas we could go ahead and apply this process on the ISS. They've actually done small-scale pyrolysis experiments there, and go ahead and be able to produce more kerosene or more rocket fuel that can then be used to further human exploration into space. And so thank you for your time, and any questions?
Thank you so much. Who has a question for Aiden?
Great presentation. You used pine for your, you said that you combined it with pine, and you mentioned that pine was a very prevalent tree in the South. However, pine was often planted by design to be a prevalent tree in the South. So have you considered any other trees, and have you also considered would the energy density be greater from actual native trees like magnolia trees and red oak and things of that nature?
Yes. Another commonly used material is switchgrass, which just grows on the side of the road in the South, really. As for magnolia, most people have a kind of a tendency not to want to cut those down because they're a lot prettier than pine trees. But really, any biomass source can contain enough energy to go ahead and produce oils from it. The most of the energy would actually come from the plastics, and combining it with the biomass would just be to go ahead and to help increase synergistic effects. And then also because when you're just throwing things away, you're not really separating your plastics from, the banana peel or something in your trash can. And so this was just to go ahead and look at a combined waste stream. Can we still produce energy from that? Does it decrease the effectiveness or anything like that?
Thank you for clarifying.
One more question.
Hi. I don't know who's going. Okay, I'll go. I go to school in Louisiana, and so I'm familiar with that longleaf pine savanna biodiversity hotspot. And so I was wondering, in terms of sustainability trade-offs, if this was to become a more common solution, is the harvesting of the longleaf pines projected to have a significant effect on the environment? Because I know there have been issues in recent years with the biodiversity decreasing in that area.
Yeah, and so that would definitely be more of a forestry kind of question, but at least for Auburn, the forestry department there, where I got all of the pine chips from, they have a pretty regular process of where they'll go ahead and cut it down, and then they'll plant new pine trees as well as other native grasses and shrubs around it after they plant that. And so the harvesting cycle is between 20 and 35 years. And so they go ahead and plant other native wildlife there as well, so that they can go ahead and continue to foster the biodiversity.
Thank you so much. So you've all been patiently sitting for an hour. Now, I don't know anything about megajoules, well, diamonds, but that's different. I don't know much about what you all talked about, but it's absolutely fascinating. And I do know that people sitting for an hour get a little edgy. So you have 3 minutes to stretch, stand up, everybody stand up, stretch a little bit. Don't be wandering the halls. Just hang out. And 3 minutes to stretch, and then we'll take resume.
You're getting pretty bold there. he already had a question lined up, and then you go in and you steal his question. Unbelievable. You need to apologize to, I forget the guy's name that had the question. Sure, yeah.
Yeah, sorry about that. I didn't know if you...
I know, you don't have to apologize.
No, he did this to spite you specifically.
I see.
I wasn't sure. I thought the person that you had gone to was who was asking the first question. I didn't realize that was I didn't know where that was coming from.
It's fine. I think he was going to go ask a question. That was cool. That was an interesting, I don't know, that was intuitive to non-experts. I like those presentations. It's like,
I was really wondering why that isn't being done. If that was, it seemed like really straightforward.
probably not a lot of money incentives to do it.
That's exciting. Very exciting. Who's subbed out? Yeah, does anybody want?
If you'd like.
I think you should take it over.
I think I should probably have it removed.
I was going to say take off your name tag so you're both Zach doing it.
That would be good. Yeah, Zach with the long hair.
You did say, why are they all on my side? It's because there's twice the number of me's.
Oh, that is actually a good point.
Yeah,
You do make a good point.
I didn't realize there were two Zach name tags.
There's also two Rishis.
There's two.
Oh, wow.
There's two Anna name tags. So it was going to be the other one. So
Wait, really? You can just give that one to me and then go and ask. I lost it. Can you?
Give it to him.
Yeah. Really, there's a lot of options. I really want just the full left side bulletproof.
I was proud my NC State friend killed me.
That's good.
She was the first in person.
Oh, okay, yeah.
That was
It's always tough going first.
1 minute left in your stretch break. 1 minute left.
I was like, I have no idea what you're talking about, but clearly you do. So
So that's good.
Was that the DNA, the supercoil thing?
Yeah.
Yeah, that was really cool.
I'm wondering how whatever was Gen R or whatever found was attracted to supercoiled segments. Is there just some sort of surface energy thing going on there? Or anyway,
Just a binding affiliation.
Yeah.
I don't know.
I did I forget. I don't think it was that one, but there was a later DNA or.
Yeah, they start to blend together, right?
Yeah. Anyway, there was one where they're probably the glucose whatever is just the smaller one, so things fit in there, and I was like, that makes sense. I get that.
The lysine thing.
Yeah.
Yes.
All right, everybody. Thank you so much.
Yeah.
Please take your seats. Welcome back. Please take your seats.
Where are you going, Colin?
Welcome back.
Okay.
Please take your seats. I am amazed that I kept you to 3 minutes. Woohoo! All right. Our next group of scholars is going to begin with Avital Isakov from Washington University in St. Louis. So welcome, Avital.
Hello, my name is Avital Isakov. I'm going to be a rising senior at Washington University in St. Louis. I'm studying chemistry, and I work in the lab under Robert Wexler with the help of Dr. Ray Yang. What we study is computational and theoretical chemistry, and because that is something that can get a little bit confusing and bogged down, I kind of wanted to focus more on what really excites me about the research that I'm doing and why I decided to join the specific lab that I joined. And that is a project we've been doing on a linear regression model for solar thermochemical hydrogen splitting, also known as STITCH. What STITCH focuses on is hydrogen fuel production, kind of similar to something we've already heard about. But what makes hydrogen fuel so cool is that it is completely carbon-free. And on top of that, it can be used to do things like offload the energy grid. So when we're producing energy through solar energy or wind energy, we actually can store that in a hydrogen bond and use it in the future, which is really exciting. And also on top of that, it is the basis for a lot of different chemicals like ammonium, which we need for a lot of different things. What I was looking at is solar thermochemical hydrogen splitting, where we basically go out into the desert, we put a bunch of mirrors out there, and we focus the sunlight onto a specific material. That material is a metal oxide, meaning it's a metal and some oxygen. And what happens when we heat it up to 1800 Kelvin, approximately very, very, very hot, hotter than anyone in this room could ever survive, we pop an oxygen out of that metal oxide. And when we do that, we can basically cool that back down, pass some water over it, and we actually can grab the oxygen out of water and produce hydrogen, which is super exciting because it's completely renewable, and we're basically recreating the material that we started off with. It's a little bit complicated, but what we can do with that is basically go out, just use the solar energy in a new way to produce a different type of energy. I work in computation. I'm not sure why that is covered weird, but a lot of times we think that computation is going to be a lot slower, bog us down in the really complicated like chemistry and the physics of everything, and we kind of think of it as the snail to the rabbit of experimental chemistry. However, in this case, we've actually been able to speed it up so that it's more a tortoise than a snail. And we've actually made a linear regression model for material design for Stitch. And what this is really exciting because a linear regression model is basically just a straight line where we can look at pretty basic things like your reduction potentials, your energy gaps, and things like the energy above the hull of a specific material that you're looking at, and basically predict exactly how much energy it's going to take to get the oxygen out of the metal that you're looking at. What makes this extremely useful is that there are a lot of different types of metal oxides, and there's a very specific range of energy that we're looking for in a metal that we want to be used in Stitch. So this model basically allows us to look through hundreds of types of materials very quickly and very accurately, and not actually have to put them specifically into like a lab setting where it takes a very long time to get through a specific material. This basically allows people who are in experimentation to handpick the types of materials that they want to spend a lot of time and a lot of money and a lot of effort on actually looking at the correct things at the correct time. And this is really important specifically now because hydrogen is at the forefront of new energy, and if it is going to become implemented in the future of like the energy landscape, it needs to be quickly and efficiently optimized. So I'll talk a little bit more specifically about my work, but I looked at a very basic metal oxide, so things like lithium, sodium, potassium oxides, and the reason for looking at those as opposed to more complicated things like perovskites, which our model was trained on, is to make this model fundamental, meaning that we know it will work for all classes of materials, and that is something that I would like to continue doing forward. And on top of that, looking at kind of using different types of programming to really make it as quick, as fast, and as effective as possible. So thank you for listening. Right on top of it.
Questions for Avital?
Hi, great presentation. So standard DFT calculations can only do a couple thousand atoms at a time maybe. Are the models that you're looking at just speeding up those calculations, or are you able to make them more complex and add in more atoms, like into the tens of thousands?
So what we look at is, I can actually show you, they're hidden, is this bottom equation is what we're actually looking at. So we make a bulk cell, and on a bulk cell we can basically figure out how many atoms we actually need in order to get to like, when we take out one oxygen, it would optimize the entire system. So we're actually looking at a minimized bulk cell. Yeah, of course.
Hey, on that linear regression model, could you explain more about the x-axis and where that equation comes from?
Sure. Can I go backwards? Maybe? There, okay. So basically what we're looking at is the actual like energy is what we're measuring. So the equation at the bottom is we're looking at kind of like 4 like pretty easily described aspects of like any metal oxide that you're looking at. And then we're kind of just trying to figure out like how, like if you like have, or if you're familiar with like a Born-Haber cycle, where you can look at things like an ionization energy or an electron affinity, and you can use that to calculate other energies that you want. We're kind of doing the same thing here, where we've actually set a model where we know like approximately what these 4 things are. If you know them, can you predict what your oxygen vacancy energy would be? So that is the use of the model. There's some like more complicated things, but that's basically what it is. You get to take like 4 simple things that are pretty easily known, pretty easily measured, and actually predict your oxygen vacancy formation energy. Yeah.
Thank you so much, Avital.
Thank you.
We'd next like to welcome Curtis Althouse from the University of Illinois. Welcome, Curtis.
Right on. All right, my name is Curtis Althouse. I go to the University of Illinois, and I study chemistry and astrophysics. And today I'm going to be telling you about the project I've been working on for about a year, which is mercury ultramicroelectrodes, and specifically how they can be used to develop the next generation of batteries, rechargeable batteries. So first, I'd like you to imagine the iPhone 20. This iPhone has 3 times as much charge, can charge completely in 15 minutes, and doesn't blow up when you hold it under a flame. Now, keep that in mind. I'm going to explain what lithium-ion batteries are and how they work, and then we'll come back to that. So, lithium-ion batteries work by having an anode, a cathode, and something in between called an electrolyte. Now, this electrolyte is really important because it has to withstand all of the potential ranges, both at the anode material and the cathode material. Now, coming back to this iPhone 20, who wouldn't want to have that? The battery industry would like to have that, specifically by increasing the charging speed, by having better cycling stability, and also by increasing the capacity. And a lot of these things, in fact, all of these things, are controlled by the electrolyte, which hasn't been changed for the past 20 years. Now, explaining a bit of the theory behind it, imagine you have a glass of water and you have a thing of salt. And as you pour salt in, it dissolves. And this is the ions, sodium and chlorine, separating in solution. Now, these ions don't just disappear. They actually form things called solvation structures, which you can see up on the right. And as you increase the concentration by adding more salt, the solvation structure actually changes. Now, the solvation structure is actually really helpful because it's responsible for all of the energy that comes from the battery and that goes into the battery in order to charge it. Now, we don't actually know how the solvation structure affects a battery, and so understanding it would allow us to develop better batteries. So the goal of my project is to develop a model to understand the kinetics and energetics of lithium-ion solvation structures. Now, if I have a model to do this, somebody could come to me and say, I want a battery that works at these temperatures, has these diffusion coefficients, and can charge this much and work at these potentials. Taking that, I could do an atom-by-atom building of an electrolyte that could meet their standards, and with that, we could develop better batteries that surpass what we have today. Now, the way that I'm doing this is with mercury ultramicroelectrodes, and these are electrodes smaller than 25 microns, so about a fourth of the size of your human hair. And with these, you can do something called amalgamation. As you scan a potential very low, electrons on the surface become very energetic, and they want to jump off and react with lithium in solution. And this forms a lithium amalgam. Now, you can do the same thing on the opposite side, which is called stripping, where you hold a positive potential, and the lithium really, really wants to leave, and it leaves this distinct curve. And this is called a cyclic voltammogram, which is not important, but I'll expand on it. So how I make these UMEs is I take a glass pipette, I thread a platinum wire through, and I seal the center. And with this pipette, I pull it in a CO2 laser puller, and I get it really, really, really small, as you can see up here. Now, this is what the surface looks like after I've polished it. And this is the 25 micron UME I was telling you about. I then etch these UMEs and recede the platinum away from the surface. And that allows me to electrodeposit mercury onto the tip, as you can see on the bottom right. And this is just a picture of a cell that I run my things in. Now, I have a lot of results, but I want to share one result specifically with you guys that I think is really interesting. So as you increase the concentration of the electrolyte, you're adding more salt into the solution, you start to see changes in the solvation structure. And as you increase the concentration, you get something called rate-limiting behavior, which you can see on the bottom left. And basically what's happening is the lithium is rushing into this amalgam, and when you start stripping it, it really wants to leave, but something isn't letting it leave. And we hypothesized that this is actually the diffusion of surrounding solvent. And this is something that has never been observed before. And if this is actually the case, it would help us understand why some of our batteries aren't working the way that we would like them to. So that's just one cool result. But as I continue to develop this model, the hope is to use this to develop new electrolytes and to expand the range of things that we can do with batteries. And with that, I would like to thank my PI, Dr. Rodriguez-Lopez, and my mentor, Abir Misha, who's a PhD candidate. And with that, I would love to take any questions that you guys have. Thank you.
Thank you so much. Who has a question for Curtis?
What do you use to polish the material, and how does that impact your margin of error?
Yeah, that's a very good question. I use sandpaper, basically. It's really, really fine sandpaper, and you basically sand the glass down until you get a flat area. And actually, the sanding allows me to get better results. If I didn't sand it, my results would be kind of not good. And so doing that actually helps me. And you can analyze these UMEs before you run them by running them in a specific solution, and the sanding helps with that a lot.
Cool, thanks.
All right, any other questions?
Hi. I might have just missed this in the presentation, so sorry if so, but how many cycles of the cyclic voltammetry are you running? And if it's a lot, why are you using that as opposed to square wave voltammetry?
Yeah, that's a good question. So I actually do use square wave voltammetry. In our literature, it's called chronoamperometry. You're applying a voltage for a certain amount of time. But cyclic voltammetry saves you a lot of time. So cyclic voltammograms, I'm running about 6 cycles, and each one shows roughly the same thing. In fact, this is six cycles, but it just saves time, and you get a lot of the same information.
Great, thank you so much, Curtis.
Thank you.
All right, our next presenter is Sydney Hummel from Purdue University. Welcome, Sydney.
Hi, my name is Sydney, and today I will be discussing engineered human artificial neutrophils exhibiting mature functional performance. So you might ask, what is an artificial cell? An artificial cell is whatever you design it to be. Artificial cells mimic biologic structures and exhibit whatever key desired characteristics of a living cell that you'd like. For my lab, we work on neutrophils. Neutrophils are a key innate immune cell, and they have desired properties that we wanted to incorporate into our artificial cells. Neutrophils are very important in the immune cell response as well as the inflammation response. However, due to their short half-life and resistance to genetic modification, we have a hard time using them in clinical applications. Therefore, our lab decided to work on an artificial version of neutrophils. The 2 properties we wanted to look at were chemotaxis and bacterial phagocytosis. So chemotaxis is the ability of the neutrophil to migrate to those sites of inflammation. And then bacterial phagocytosis is a way for it to combat those infections. My project overview was to first start with genetically engineering a K562 cell line, that is a human leukemia cell line, to express a GPCR of interest. GPCR is G protein coupled receptors. These had been shown previously with neutrophils to be a part of the chemotaxis process. Next, I worked on encapsulating vancomycin-loaded silica nanoparticles. The goal of these was to aid in the bacterial phagocytosis of our artificial cells. And then finally, we evaluated the performance of these artificial neutrophils. So first, like I said, the K562 cells were genetically engineered to express the RG-PCR of interest. As you can see from the diagram here, the K562 cells are the original cell line, non-genetically edited. And then whenever you see the HM4DI K562 cells, you can substitute in artificial neutrophils. So that red fluorescence there shows the success of the genetic editing for these neutrophils to express the desired receptor. We were able to show that neutrophils do demonstrate enhanced chemotaxis. So the artificial neutrophils were able to migrate across a trans-well barrier with the use of CNO, which is an inert drug that was designed for the receptor. We also were able to see that they do have the enhanced bacterial phagocytosis abilities as well, with our artificial neutrophils showing similar percentages of bacterial phagocytosis to natural neutrophils. This technology serves as an avenue to investigate neutrophil properties in vitro, whereas you might not be able to do that, again, due to the short half-life of these cells. There are clinical applications in targeted therapeutics and disease treatment and prevention. Further research needed to be done on this subject. It was a proof of concept study, so we would like to look into the potential cytotoxic effect of using a tumor cell line as a source cell line, as well as investigating potential neutrophilia that may occur based on this technology. And finally, I'd like to thank my advisor, Dr. Xiaoping Bao, and my advisor, Dr. Yun Chang, as well as funding from the Davidson School of Chemical Engineering at Purdue, the National Cancer Institute, and then the Astronaut Scholarship Foundation. I'd be happy to take any questions now.
Wonderful. Who has a question for Sydney?
Hi, Sydney. Great presentation. Finally, something I can ask a question on here. So I'm just curious, have artificial cells been proof of concept tested in humans or animals, not just neutrophils, but any other variation?
So I do know that artificial cells have been generated for T cells as well. We looked at neutrophils specifically because of their migration ability across the blood-brain barrier. In that way, I'm not entirely sure if that has been used in humans, and it's something that I'd like to look into. So yes, thank you for your question.
Hi, Sydney. Great talk. Could you expand more on how you determined that these artificial neutrophils had a better ability to do bacterial phagocytosis?
Yes. So what we used was a fluorescence way to measure this. So the bacterial particles were tagged with the green fluorescent protein. And so when the cells were able to take up the bacteria, we were then able to measure which cells expressed the fluorescence versus the cells that did not. And this was measured using flow cytometry, which is shown here.
All right. Thank you so much, Sydney. Our next presenter is Audrey Rips Goodwin from the University of Kansas. Welcome, Audrey.
Alrighty. Thank you everyone for being here today. I'm going to discuss how I used chemistry and psychology to examine the accuracy of reported energy content for hyperpalatable and non-hyperpalatable foods. So to begin, I'm sure you are all wondering, what is a hyperpalatable food? Well, here we have Oreos, potato chips, and pepperoni pizza. Now, what do all of these foods have in common? They're both delicious and difficult to stop eating. I mean, who can have just one potato chip? These are hyperpalatable foods, those that override physiological satiety mechanisms, thereby leading to overconsumption despite physical fullness. So you might be physically full after eating these foods, but your body's not satiated and you're not satisfied, so you keep eating them. And this is not by chance. Hyperpalatable foods are specifically formulated to induce a highly rewarding eating experience. And this is very important for all of us here today, as hyperpalatable foods comprise 60% of foods in the US food system. So, what would happen if hyperpalatable foods contained more calories than they said they did on the label? Well, excess calories may hinder population weight management efforts as people are consuming more calories than they think they are, and these foods are difficult to stop eating, so that kind of exacerbates this. Further, this would serve food companies' interests as people are consuming more of these already addictive foods and thus buying more of them. So this leads to my driving question of, do hyperpalatable foods tend to vary more from their reported energy content on their label than do non-hyperpalatable foods? So, in essence, I'm asking, does this hyperpalatable Nature Valley bar vary more from its label and its reported energy content than does this non-hyperpalatable Kind bar? So, in this study, excuse me, I selected 38 foods in a matched pairs design. So I had 19 hyperpalatable foods with 19 non-hyperpalatable comparator foods like we saw in the previous slide. I selected a wide variety of foods across the US food system, mostly focused on packaged foods. So to determine the energy content in the food, I used a bomb calorimeter. So this means that I took the food or the fuel in this equation, I put it into the bomb and I blew it up, because this is a combustion reaction. And energy was released in the form of heat, which changed temperature in some water. And using this change in temperature, I determined the energy in this food using this equation that we know from general chemistry. And I then calculated the kilocalories per gram and compared that to the label. And so in the end, hyperpalatable foods did not tend to vary significantly more from their label than did non-hyperpalatable foods. So there was no meaningful difference in variation in the reported energy content. To do this, I determined this using a Wilcoxon sign rank test with a 95% confidence interval, which is a non-parametric statistical test analogous to a t-test that compares the median of two groups. So we can see that the hyperpalatable foods and the non-hyperpalatable foods had very similar medians, and thus the data were centered in a similar area, and there was no significant difference. However, overall, there was a pretty wide range and variation from the label for all of the foods, ranging from about 1% to 84%. So in conclusion, consumers should be reassured about the labeling of hyperpalatable foods compared to non-hyperpalatable foods because they do not vary significantly more. However, foods may contain more calories than reported on the label, and this variation may depend more on the type of food rather than the hyperpalatability of the food. And once again, this impacts population weight management efforts as people may be consuming more calories than they think they are, which may impact their calorie counting measures. In the future, I'd like to further examine the variation by type of food. For example, looking at dairy versus snack foods, why one may vary more from its label than the other. And in my future project, I will be analyzing the sugars in infant formula as it pertains to childhood obesity. Thank you, and I'd like to say thank you to my research advisor, Dr. Tara Fazzino, my collaborator, Dr. Lisa Sharp-Ellis, and Professor Misha Barabin for letting me use some stuff in his laboratory, and for the Kansas Idea Network for Biomedical Research, an NIH-funded program for giving me funds for this project.
Thank you. Questions?
Did your variation tend to be an overestimate of calories or an underestimate?
There was, I don't know off the top of my head the exact number, but there tended to be, there was both for sure, overestimation and underestimation, but it was definitely more overestimation. So it varied. There are different types of hyperpalatable foods. There are 3 clusters of those, and so it varied on the type of hyperpalatable food, but more so I saw more variation in the type of food, like the category of food. So I saw a lot of variation in dairy and meats. Maybe those foods weren't fully dehydrated and there was an error there compared to more like snack foods, but I'm not really sure where that variation is coming from.
How did you make sure that what you were putting into the bomb calorimeter was homogeneous? So like, for example, the Kind bar, how did you make sure that you got a full representative sample of that bar when you put it into the calorimeter?
Yeah. So I dehydrated the foods in a vacuum oven dryer at a very low temperature, and then I homogenized them using a coffee grinder, which is, I know, very high tech, but they were just pulverized basically and made into a compact pellet to then go into the bomb.
Great. One more.
Great presentation. Were there any other classes of food or subgroups that you saw that did exhibit a statistically significant variation?
That did or didn't?
Did.
So I also, this was not in this statistical analysis, but I did have like fruits and vegetables and some more like natural foods like that, and those were all over the place due to the fact that we can't kind of control for things going on when those foods are being grown, so we don't know the exact amount, and then water, and there was just some weird things that I'm not really sure what was going on with them.
Thank you so much. I truly have been wondering for a very long time, what is going on with Oreos? But that aside, so our next presenter, let's welcome Carly Lebendorfer from the Florida Institute of Technology. Welcome, Carly.
Hi everybody, my name is Carly. I am a rising senior at Florida Tech. The past year and a half, I've been working in the Functional Biomaterials and Tissue Engineering Lab. And this summer, I pretty much got to play with magnets. So a little bit of background, each year there's roughly 175,000 knee injuries that result in an ACL reconstruction. This is commonly done by using an autograft, which will take some tendon within the patient's body, like the hamstring, and use it as a graft for the ACL. Our lab is looking at creating a novel scaffold system which aims to regenerate the tissue by using cells in the body and a porous biomaterial. So we want to influence a functional regeneration of the ACL emphysis. Now, the emphysis is the part of the knee that connects the bone and ligament, and particularly with implants, this is a high area for failure. So some personal motivation, I'm a current collegiate athlete, and the ACL has, especially for females, a high rate of recurrent injuries, especially in a contact sport like lacrosse. So some methodology. The purpose behind this whole experiment is to induce a high degree of collagen alignment using magnetic particles that, when placed parallel to a magnetic field, will themselves align and hopefully influence the collagen to align. This is important because in the native ACL environment, you are seeing a high degree of collagen alignment. So to begin our experiment, we create a hydrogel using collagen neutralizing solution, a photo cross-linker, and small magnetic particles. Then we can do two things. We either first will place it into an incubator, or we can place it under the magnetic field, or we do a variation of both, with time being our independent variable. Once they have both been incubated and magnetically aligned, we will then place it under a UV light to expose it, which enhances the material's properties. From there, we then can begin the sample preparation and drying process to image it using a scanning electron microscope. From these SEM images, we then can do some computational analysis to really quantify the alignment that we're seeing. So some results from these SEM images, and we've run different experiments. These first images are, we have our hydrogel and our magnet placing on top. So what we're seeing is a high degree of alignment when placed under a magnetic field for an hour and then incubated for 30 minutes. This was great. We loved seeing all the alignment. The downfall, if you look at the image with 1500 magnification, the underlying collagen isn't being aligned. So this is a little bit of an issue. However, when we place the hydrogel under the magnetic field for 15 minutes and then incubated for 30, we're seeing that random orientation of the collagen and the magnetic particles since they haven't had enough time to align. We could also see this visually with the top one, as the magnetic particles are brown and your hydrogels are clear. So visually, when we're looking at them after they've been incubated, we are seeing a layer of brown and then clear, which makes sense because if a magnet's on top, you would expect the magnetic particles to kind of rise. So from here, a main bulk of my project has been the alignment quantification. So at the beginning of this project, I was really trying to learn how to MATLAB code this, and coding is not my expertise. So I really dug into the literature to see somebody's had to do some kind of collagen alignment, and I've actually found 2 MATLAB programs that use FFT, which is a fast Fourier transform, and that was Cytospectra, and then another MATLAB-based program called Curveline, which does a curvelet transform. So we were able to find 2 programs that would give us the same result of alignment, which was great because we could really confirm our results. So we could look at values like circular variance and circular skewness, which would tell us a little bit about the alignment that we're seeing with that underlying collagen. So what are our next steps? So in the end game of this, we would like to create a material that we can then 3D print into our ACL scaffold, place under a magnet and magnetically align it to get that ACL-like collagen fibril alignment. And then of course we would introduce different kinds of cells and growth types, growth factor types to really hone in what you're seeing in that native ACL environment. So we just got this funding for this grant project in April, so we are in the very preliminary developmental stages marked by that green X. We have a long way to go, and our experiments aren't really working right now. So we definitely have a lot more troubleshooting and research to do. I want to say thank you to ASF for allowing me to speak today, as well as my lab and my Dr. Kishore, who's my PI, and Nash, who was the PhD student that I was working under. Thank you.
Thank you. Who has a question?
Is there a reason that you're using magnetic particles as alignment techniques rather than things like electrospinning, which would allow for forced alignment of the fibers, rather than introducing like things that are not regularly found in the body?
Yeah, so you definitely can achieve alignment through things like ELAC and all of that, but we're trying to step out of what's being done right now to find a different approach, and magnets are cheap and pretty easy to use. So if we could find like maybe an alternative to something like ELAC that requires like electricity and a little bit more of a setup, it's pretty much like that mass reproducibility. Like what's the easiest thing? And 3D printing is something that's cheap, fast, and easy to use. So if we can print these scaffolds and then put it under something like a magnet, it makes it super easy and super fast to mass produce something like this. So that's kind of like the end goal in mind.
Another just small thing, ImageJ has an orientation analysis for fibers that you could also use.
Yeah, I tried using that and couldn't figure it out.
All right. Thank you, Carly. Next, we want to welcome Eric Roche from Johns Hopkins University. Welcome, Eric.
Alrighty, hello everyone. My name is Eric Roche. I am a biomedical engineering undergraduate student at Johns Hopkins University, and I'm excited to tell you all about some of the work we're doing in the Green and Sunshine labs there on polymeric nanoparticle gene delivery. So we'll talk a little bit about gene therapy broadly, and then some of the specific work that we're doing now. So what is gene therapy? So gene therapy involves the delivery of nucleic acids like DNA or RNA to the inside of cells to see some therapeutic benefit. And that benefit could be very broad. It could be in autoimmunity, in cancer therapy, in vaccines, or even regenerative medicine. And this might seem super futuristic, but most of us have actually received gene therapy very recently with the new COVID-19 mRNA vaccines. And so with all these applications, you might be wondering, why isn't gene therapy everywhere? And so it was once said there are 3 problems with gene therapy. 1 is delivery, the 2 is delivery, and 3 is also delivery. And so you might be wondering, why is it so hard to get DNA or RNA to the inside of a cell? And so the reason for that is that DNA is extremely negatively charged, and it can't cross into the inside of a cell. And so the obvious solution might be to use a virus. So a virus is extremely efficient at getting DNA to the inside of your cells. And using an inert, non-pathogenic virus is actually proven to be very efficient at this. However, there's a lot of drawbacks. Making non-pathogenic viruses for this purpose is extremely difficult to do, and that makes them extremely expensive. And just like viruses have millions of years learning how to efficiently get DNA to the inside of your cells, so your body has millions of years learning to fight them. So you often can only get 1 dose out of it before you build an immune tolerance, and the therapy no longer works. So to drive this point home, this is a current FDA-approved viral gene delivery that costs $2.1 million for just 1 dose. And so this really inspired the work in our lab to use polymeric nanoparticles to deliver genes to the inside of cells. And these have a lot of really exciting benefits. We're virtually unlimited with the size of the genes that we can deliver. They're very easy to manufacture and quality control, and there's no natural immunity against them, so you can get multiple doses. And so the specific polymers that we use are a polymer called a polybeta amino ester, or PBAE. And these have some really exciting properties I'll get into now. So I mentioned how that really strong negative charge in DNA and RNA allows it not to cross into the cell. So our polymers are actually positively charged. So picture a nanoscale, positively charged piece of spaghetti. And so when you mix it with DNA, it actually self-assembles into these tiny nanoparticles, way smaller than even a cell. And that shields that negative charge in the DNA and allows it to enter super efficiently into the cell, into a vesicle called an endosome. Then the polymer is actually able to buffer that endosome, disrupt the membrane, and then escape into the cytoplasm of the cell. Once it's in the cytoplasm, the polymer is actually completely biodegradable. It degrades in water. So then it's able to release that genetic cargo into the cell. If it's RNA, it can go right to work. If it's DNA, it's then shuttled into the nucleus where it can be expressed and we can see that therapeutic benefit. So what's the specific work that we're doing now? So our generation 1 canonical PBA polymers have this linear structure. That's that positively charged piece of spaghetti I mentioned earlier. So we thought, what if we change the chemistry slightly to get this 3D branch structure? So picture roots of a tree in this complex 3D network. And so our thought is that could allow the polymer to complex more efficiently with the DNA and allow it to enter into the cell more effectively. And so we've seen some really exciting results with that. This is an image of some melanoma cells plated in a dish that we transfected with nanoparticles that code for a protein that turns them red. Or sometimes we turn them green, depending on the application. And this allows us to understand how effectively our particles are working. And so the way this project works largely is we synthesize a library of polymers. Today we synthesize 70 that have this kind of branching structure. And we test them in a dish. We turn the cells red or green, depending on the application. And we use that to synthesize a new round of polymers to continuously optimize the structures that we're using. We also characterize the nanoparticles we form, look at the size or the charge on the surface of the particle, and then test them in an animal model to make sure we're seeing that therapeutic benefit that we want. And so the applications of this we're really excited about are in cancer therapy and immunoengineering, which are the specific projects that I'm involved in, and also retinal gene delivery, which is delivering genes to the cells in the back of your eye. And I'll wrap up here in a second. I just want to thank my incredible mentors. Catherine Lulai is my graduate mentor, as well as my PIs, Dr. Zhang, Sunshine, and Green. And also to ASF and all the scholars for just an incredible week here. So thank you. I'd love to take questions.
Thank you, Eric. Questions?
Forgive me, because what I do is nowhere near biochem. I was wondering, since you said that PBAs are even dissolvable in water, how do they get to the cells in the first place?
Yeah, great question. So I'll kind of go back here in a second. So we have these polymers that are actually stored in organic solvent on -80, and that prevents them from breaking down. Then we'll mix them with the genes that we want in water, and then that forms the nanoparticles, and then the clock is ticking. So to store them right now, we'll put them on -80. Sometimes you can put them in a freezer. We're looking to try and stabilize them. Then when it's ready to inject them, for example, in an animal model, we'll thaw the polymer, and we have a couple hours before they would have to be injected. Once it's in the cell, we're talking about a couple hours before it'll degrade. And once it's actually injected, it absorbs very fast. So then once it's inside the cell, it'll break down. Yeah, great question.
Go ahead, one more.
Is there anything that you're doing to kind of increase endosomal escape? Because I know that especially DNA nanoparticles and things like that have a very, very high tendency to be stuck in lysosomes and endosomes as well.
Yeah, that's a super awesome question. So I'll skip to the structures we have here. So there's 3 colors on this structure here. The red is the backbone, the blue is a side chain, and the green is an end cap. And you'll see all these ends that are mixed in here. They're called amine groups. And so that is actually what allows the polymer to buffer the endosome and escape. The way it does that is something called a proton sponge theory, which is still debated on exactly how that mechanism works. But this 3D branching structure allows us to get a lot more end caps on the polymer, which are those green sections, which means we're able to buffer more strongly. And it's actually a quality that we've seen that is necessary for transfection, but not sufficient for transfection. So it was a great question.
Wonderful. Thank you so much,
Thank
Eric.
you.
We'd next like to welcome Chloe Breckus from Colorado State University. Welcome, Chloe.
All right, so I'm going to be presenting osteosarcoma fracture risk prediction using finite element modeling, which is a type of computer modeling. And so oh my goodness, let's restart that.
In cancer patients, pathological fractures due to osteosarcoma or other bone tumors are a common problem. And right now, clinicians look at retrospective analysis of similar tumors to decide what fracture risk the patient is at. And this lacks a lot of patient specificity because it's not looking at exactly what their tumor is, and these tumors can vary a lot from patient to patient. And so our goal was to develop and validate a patient-specific finite element technique that can predict fracture in cancer patients better than current techniques. And so for this project, we used a canine model. So we collected canine cadaveric radii. We took a semicircular osteotomy out of the bone to simulate a tumor resection. And then we took a computed tomography scan of this bone. And then to validate this model, we did both mechanical testing and finite element modeling and compared the results. So we pretty much want to see that the model is pretty much the same results as the mechanical testing, so the bone is performing as you would expect in real life. So first we started with mechanical testing. So here you can see the samples were loaded in an MTS. We did 3 samples in torsion, 3 in bending, and then 2 in compression. So all samples were loaded to failure, and then we took stiffness and yield load data from these. And then for the finite element modeling, we started with the CT scan prior to mechanical testing. We segmented it to create a 3D model that exactly replicates the bone geometry. Then we created this finite element model, so we made a mesh, so there's a ton of tiny elements in this bone. And then we assign material properties using this equation. And so what this equation does is it pretty much takes the bone density from the CT scan and turns it into an elastic modulus so that each of these elements has a unique material property as you would expect in an actual bone. And then finally, we applied loading to replicate the mechanical testing setup. So we either applied a bending load, torsion, or compression. And then we calculated yield load based off equation 2, which uses the applied load and the maximum principal strain. And so here are the results. So in the top row is stiffness, bottom row is yield load. And you can pretty much see that the darker is the experimental or mechanical testing, and then the lighter bars are the finite element. And what we wanted to see was that both of these bars should be pretty much the same for each of these samples. So each set of bars is a different sample. And as you can see, the model very accurately predicted stiffness since all those bars look pretty good. And then for yield load, there's a little more variation going on, so we're currently working on modifying that equation 2 on the last slide to get more accuracy in this regards. But we did statistical analysis doing paired t-tests comparing experimental versus finite element and didn't find any statistically significant results, which supports the validity of the model. And so in the future, we would use this clinically to provide quantitative data to clinicians that can assess fracture risk. And so you can look at different physiological loads that the patient's limb might undergo in their daily life and compare this to the yield load that predicted by the model. And based off this, you can decide a fracture risk, and then these clinicians can make better treatment decisions for their patients. So, for example, if you have a really high fracture risk, you might need surgical intervention to stabilize the bone or amputate the limb entirely. And if you have a low fracture risk, you can be spared unnecessary surgery. And so in the future, we'd like to validate this on bones with actual osteosarcoma lesions. So in the picture to the right, you can see a model I'm currently working on that has a pretty big tumor at the end. And what we need to do with this is make sure that our material property equations still work for the bone tumors, and that we may have to change it because those equations are specific for normal bone. And another thing we can do with these models is test different types of internal fixation. So I mentioned that would be one of the treatment factors that you would look at with these patients. And so you could look at if you put like a rod through the bone, or if you need to put screws through the bone to make sure it doesn't fracture. That's different stuff you can test with this model. And eventually we'd like to translate this from a canine model to human patients. And so I'd just like to thank my lab, the OBRL at Colorado State University, and my PI, Dr. Godomsky. Thank you.
Thank you, Chloe. Questions?
Do you plan to include any metrics in your models for like bone health or like the age of the patient?
So yes, so the beauty about this model is that because it's patient specific, you don't necessarily have to take those factors into account. This uses the exact bone density from the CT scan, and so that accounts for different factors like age and gender. So yeah.
Hi, thank you for your presentation. I have a question about the structure of the bone. Since I imagine bone is heterogeneous and there's
Yes.
lots of density gradients within the material itself,
Yep.
does accounting for that like gradient and differences, let's say between the elasticity on the surface versus volumetric elements, would that greatly differ from the type of modeling you have used?
Yeah, so with this model, both cortical and trabecular bone are captured by the equations we use. And then the center of the bone is different. It's the medullary cavity, so there's a lot of bone marrow in there. And so we use just an isotropic elastic modulus for that section, so it's a little different. But for the actual bone tissue, it's all captured by that equation, if that answers your question.
Thank you very much.
Thank you.
Next, we would like to welcome Ronan Joshua Kosikin from the University of Minnesota. Go Gophers.
Yeah, go Gophers. All right. Hi, everyone. My name's Ronan Joshua Kosikin, and I'm from the University of Minnesota. I'm a rising senior majoring in genetics, cell biology, and development. Today, I'll be discussing a case study about a patient who had Anaplasma phagocytophilum who presented with encephalitis. I co-wrote this case study with the following doctors, and I'd like to give special thanks to Dr. Igor Dumich, who from the Mayo Clinic Health System, who was my PI on this case. So after having spent a few days together here in Florida, I'm sure we've all gotten our fair share of the heat and the rain and maybe the mosquitoes. Where I'm originally from, the state of Wisconsin, we're pretty much known for our abundant supply of cheese and our cold winters. But during the warmer months of the year, we have a different insect instead of mosquitoes that likes to come out, and those are ticks. Ticks are vectors for a number of different diseases. We've all probably heard of Lyme disease, which is one of the most common vector-borne diseases in the United States. But other tick-borne diseases include babiosis, ehrlichiosis, and the disease I'll be discussing today, which is anaplasmosis. Now, before I jump into the details of the case study I'll be discussing, I'd like to go over some basic information about anaplasmosis. Anaplasma phagocytophilum is the bacteria which causes human granulocytic anaplasmosis. The disease is classified as granulocytic as it affects the white blood cells in your body. As I previously mentioned, it is transmitted via tick vector, primarily the ticks Ixodes pacificus, otherwise known as the western black-legged tick, as well as the Ixodes scapularis, pictured here, known as the deer tick or just the black-legged tick. The disease has a number of symptoms, including fever, nausea, headache, muscle aches, and is typically treated with the antibiotic doxycycline. Now that I've discussed some general information about Anaplasma phagocytophilum, I can go over the case presentation. The patient was a 62-year-old male who appeared in the emergency department with confusion, while his wife reported that his speech was becoming increasingly incoherent. He had a number of different symptoms, including headache and a 38.4 degrees Celsius fever. He also reported that he enjoyed spending extended periods of time outdoors and had previously removed a tick from his skin around 4 weeks prior to his appearance in the emergency department. After he was admitted to the hospital, he was given a treatment of the antiviral acyclovir and the antibiotics ceftriaxone and vancomycin, but his fever continued to persist. To further investigate the causes of the patient's symptoms, a number of tests were ordered. The patient's complete blood cell count, urine analysis, chest x-ray, and MRI all came back normal and showed no signs of abnormality. For the physician in charge and the attending physician, they thought that the patient's symptoms of fever, confusion, and headache suggested encephalitis, or inflammation of the brain. To investigate this, they ordered a lumbar puncture, which is otherwise known as a spinal tap, and the collected cerebrospinal fluid was then sent to the laboratory for testing. The spinal fluid was tested for a number of different diseases, some of which are shown in the panel here, and specifically was tested for tick-borne illnesses. The patient came back positive for Anaplasma phagocytophilum, highlighted by these gold boxes, and he was immediately treated with the antibiotic doxycycline, the typical treatment for anaplasmosis, and within 24 hours, his symptoms had improved, and he was able to be discharged the following day, under orders to finish the remainder of his 2-week cycle of doxycycline. When the patient returned for his 3-month follow-up visit, he had no residual symptoms and was fully recovered. Now, why is this significant? Well, neurological manifestations of anaplasmosis are quite rare, and this is actually the first ever reported case of a patient with anaplasmosis having encephalitis. So this further brings to attention the need for increased research into the relation between Anaplasma phagocytophilum and neurological issues. Speaking more generally, since most of us probably don't think about ticks on our day-to-day, I think that this case study and case studies in general are great examples of how we can bring rare cases in science and medicine to the public knowledge, and they demonstrate that there's always more to be learned and more to be studied in the fields of science and medicine. So these are some of my references, along with the reference for our case study, and thank you very much for listening.
Thank you very much. Questions? Not one. Oh, there we go.
Do you know where the tick bit on the body, and do you have any idea if that affected how the disease transmitted throughout it?
I'm not sure. Based on our report in the case study, he didn't really say where the tick was, but I didn't personally see the patient. It was my PI that did, so I can't really speak to that.
Really cool presentation. You said that the complete blood count was normal. Is it normal to not see an elevated white blood cell count in patients with this?
You'd kind of expect, because there's an infection, that the blood cell count would be abnormal, but that's kind of part of the reason why we decided to report it, because we thought that was weird and interesting. So.
Yeah, that is interesting.
All right, well, thank you very much. No ticks. No ticks. Ticks are bad. Since I live in Minnesota and have dogs that have long hair, and ticks love that, so great, very interesting. Okay, Brian Dong. Welcome, Brian, from Johns Hopkins University. We're going to take a break.
See how this works. Okay, hello, good afternoon. My name is Brian Dong. I'm a rising senior at Johns Hopkins University studying neuroscience, and I will be presenting on one of the projects I've completed in the past year. And the background of the nervous system is basically, it's the way that humans can interact with the environment and perceive the environment. And the cells in the nervous system are neurons, shown here, and they connect to each other with axons. So axons are these very long, relative to the size of the cell, very long projections that allow neurons to communicate over distance. And a hallmark of neurodegenerative and neurotraumatic diseases is axon destruction and axon death, and this will lead to retrograde tracing until it reaches the actual soma or cell body of the neuron, and the soma itself would die. So this is very common in Alzheimer's, spinal cord injury, and glaucoma, and it leads to around, I think the number is $100 billion in medical cost per year, so it's a very challenging biomedical issue. And my project is, how can we stimulate the central nervous system to regrow axons after insults? And the molecular mechanisms of this is that the central nervous system will transition from a high to low growth state across neurodevelopment. So basically, when DNA codes for proteins, DNA is wrapped around these proteins called histones, and the tighter it is coiled, the less transcription can occur. So this is low coiled DNA. This is very tightly packed DNA around histones. And when the central nervous system, or CNS, develops, it goes from a state like this to here. So we have decreased transcriptomic activity across neurodevelopment. And when we want to regenerate axons, we want to stimulate a lot of processes like metabolism, cell growth, and that requires a lot more loose of a configuration in the histones. And what I investigated was a type of transcription factor called a pioneer transcription factor. And PTFs, like, let me see, this one here, can access these closed histone states to recruit more open complexes for transcription to occur. The model I used was the retinal ganglion cell injury model. So basically, retinal ganglion cells, or RGCs, are neurons that line the inside of the retina, and they transmit visual information into the brain through the optic nerve. And the optic nerve can be crushed to simulate injury. And before we do the crush, we actually inject a viral construct that has the gene that we're trying to knock down or upregulate into the eyeball or the vitreous chamber. And once that gene is expressed, then we do the optic nerve crush, and then we also wait for that to regrow and inject the fluorescent tracer to visualize growing axons. And the timeline's roughly a month. And what I found specifically was this gene called ASCL1. ASCL1 is a pioneer transcription factor that is known to direct neurogenesis and neurodifferentiation during development. And based on some very large sequencing analysis of public data sets, namely single-cell RNA-seq and ATAC-seq, we could find that ASCL1 was a very potential regenerative factor. And I did the ASCL1 overexpression experiment, resulting in this on the right. So you can see a lot of axons here that are regrowing 14 days after injury. And at all lengths, it was very statistically significant. And for future directions, we want to see if we can reprogram nervous system to regenerate axons with more than one factor, so ASCL1 with other transcription factors. And thank you to my mentors and the ASF Foundation for providing me funding.
Thank you, Brian. Who has a question? There's one in the back.
Yep.
Hey, Brian, nice job. I actually have two questions. My first one is, I don't remember if optic nerve injury models create glial scars the same way that a spinal cord would. So that's question one. And question two is, did you also look at these new axons? Were they successfully remyelinated as well?
Yeah, so great question. Going to the glial scars, I believe they do, but recent work in 2021, I think, shows that even if you like kind of erase like the extracellular matrix damage, there's still an intrinsic problem with mature CNS cells and why they don't regenerate. So even if you get rid of like the scarring, the glial scarring, as you say, there still needs to be some sort of cell autonomous response. And the second question, remyelination, that's a very interesting topic. I'm not sure if off the top of my head, I'm not sure if there's a way to find that out in the optic nerve, but yeah, that's a very good point in terms of the process to functional recovery that I can look into. Thank you.
Thank you. One more question. Anybody have another question? All right, well, thank you, Brian.
Thank you.
So because our scholars are brilliant, number one, and because they are right on time, you get not 2 minutes, not 3 minutes, but 5 minutes. Be back in 5 minutes, please.
last year I remember being like more just like separated
Yeah, it's like all the space ones were together, all the biochem ones were together. Quantum computing, they were like,
yeah. Well, I guess a lot of the space ones would be.
Yeah. Yeah, it's interesting. Like, I feel like
I was reflecting on how much of a policy background there is. You have it, like, within biology, like, see biology, and you're like, oh yeah, GPCR, we do that in there. It's like,
I like the ones that are like trash plastic. Or it's like plastic gas or whatever.
that was But I'm just curious first with like when you guys do like space, like you do it just super wide, you know. Maybe I thought I saw one for tomorrow, but maybe I was wrong. Oh, yeah, okay, I was going to say, yeah, that's what I saw, the Arctic air sea fluxes. Oh, really? Oh, that's interesting. Sarah Samers is coming from my university. Her talk is just like, not like super technical. I love it. I'm at a big science on there. It's good stuff. Don't worry about you guys getting together. Yeah, we're conspiring to like, follow the presentation.
UVA, that's interesting. I didn't know they had weather.
All right, everybody, 5 minutes is up. Please take your seats. Please take your seats. Hello, take your seats, please.
Sure, yeah. All right. Yeah, of course.
You're in place. I love that. That's awesome. You threw a curveball at me by adding a break.
That's awesome. Again, I'm thrilled. We were at 5 minutes and you're all here. Yay! So, we're in the final stretch. We have 8 more presenters, and we're very excited about all of them, so hang in there, and we're perfectly on time. So, I don't know about you, but I'm fascinated by every single thing I'm hearing and learning so much. And, you know, if Kurt and Mary just said, and myself too, I'll add to it, if you need ticks for experiments, come to we got a lot of that going on. So please welcome Kayla Gapin. Did I say that right?
It's Kayla Gapin.
Thank you. And she is from the University of Colorado Boulder. So welcome.
Thank you. So yeah, my name is Kayla Gapin. I'm a rising senior at the University of Colorado Boulder, and today I'll be discussing the research I performed over the summer at the University of California, San Francisco on the three-dimensional reconstruction of nuclear topography in the context of this very special protein, in my opinion, called air. So a little bit of context and background on the immune system. One of the primary organs in the immune system is called the thymus. It's located about here, and it has 2 compartments, the cortex and the medulla. Together, they're involved in the selection of T cells for the proper development of the immune system. T cells are one of the most important cells in your adaptive immune system. And so together, the cortex and the medulla make sure that they perform their function correctly before they're released out into the rest of the body. The cortex is used for selection of, I'm sorry, the slides are going without me pressing the button. The cortex is involved in selecting for T cells that are reactive to pathogens, so you want your T cells out in your body to actually elicit an immune response when you've been infected with something. And then the medulla is involved in making sure that those T cells are not also self or autoreactive. So those same T cells that you want eliciting an immune response in response to pathogen, you don't want eliciting that same immune response in response to your own cells. And the way that the medulla does this is through this particular cell type called MTECs, so medullary thymic epithelial cells. All of the cells in your body have the same genome, but each cell type expresses a certain subset of those genes. So your muscle cells produce different proteins than your neurons, and that's a very good thing. They're different cells, they have different functions. But we want to expose our T cells to as many self-proteins as possible in order to train them not to respond to your own cells. And the way that MTECs present all of these typically tissue-restricted antigens is using this protein called AIR. AIR is the autoimmune regulator. It's pretty aptly named. If you have certain mutations in AIR, you tend to develop autoimmune disorders. And we know that it's a transcriptional regulator. It tends to globally upregulate transcription in MTECs. So as a population, MTECs, and this is very unusual, together they're able to express over 90% of all of the protein coding genes in your genome. But there's a lot that we don't know about AIR. And so this is still a very heavily researched topic. We know that AIR seems to release stalled RNA polymerases, and it's not a typical transcription factor. So it doesn't bind to specific sequences in your DNA. Rather, it's being localized to regions with certain histone modifications, which you've heard about earlier. So it localizes to areas of closed chromatin to help open them up. One of the other things we know about AIR is that it forms these subnuclear structures, so these nuclear bodies that we can visualize by microscopy. We don't know a lot about these bodies. Some of AIR's protein binding partners are known, but not all of them, so we know some of the proteins that might be in these areas. We know nothing about the RNAs that are in these bodies, and we know that AIR tends to localize and loop super enhancers towards other promoters and genes in the genome to help increase expression of these genes. But generally speaking, we don't know what DNA is found in these speckles. We know that they're not typical or well-characterized subnuclear structures like the nucleolus or PML bodies, and so these are really distinct and we don't know much about them. So this summer, I was aimed at studying these nuclear bodies that contain air, and the way that we did this was using this protocol called SPRITE. It's pretty novel, and my role was to establish this protocol in the Anderson and Gardner labs where I worked. If you're familiar with high C or 3C techniques, SPRITE is an adaptation, and I would say perhaps an improvement on those. It gives us a little bit more information. And so typical techniques for 3D reconstruction of nuclear topography are based on proximity ligation, and they're pretty limited in that scope. And so the difference with SPRITE is that we can look at a different scale. So we can look at things like nuclear speckle formation. The general idea behind SPRITE is that we fix all of the proteins and the DNA in the nucleus to one another, and then we break the chromatin apart into these clusters. Each cluster then gets a very unique and specific barcode, and when we sequence these libraries using that barcode, we're then able to deconvolute all of the pieces of DNA that are interacting within this given cluster. So all of the Why is it doing this? All of the DNA that is interacting with a given protein or protein complex. And the way we're adapting this to air is we're adding this specific modification that hasn't been used with SPRITE before. So I generated an oligo-tagged anti-air antibody. And the idea is that when we introduce this into this protocol, the antibody will bind to air in the nucleus, and then that oligo is compatible with the rest of the protocol such that it receives the same unique barcode as all of the rest of the DNA in a cluster. And so when we sequence this particular library, we not only get information about what DNA is involved in is interacting in a cluster, but also whether that cluster contains air. And so we've sent a library off for sequencing. Unfortunately, that takes a while, so we don't have results back yet. But this is the type of information you can get back from a SPRITE experiment. When we look at the data at different resolutions, we can start to explore things like A and B compartments, so transcriptionally active versus inactive regions of chromatin, and even down to looping scales. And for our purposes, we're interested in looking at how the chromatin is being remodeled at a nuclear speckle or subnuclear structure that's visualizable by microscopy, for instance, scale. So in the future, we're interested in performing single-cell SPRITE, so starting to explore the variation between MTECs and how air is being expressed in those cells. We know that air expression and the expression of certain tissue-restricted antigens is pretty variable between MTECs. I mentioned earlier that as a population, they reach 90% of all of the protein-coding genes, but that's not all of the same genes and all of those genes in all of the cells. So it's interesting to look at a single-cell level what that can do. We're also interested in using a protocol called SPIDER, an RNA-DNA SPRITE, to start to explore what kinds of RNAs are in these speckles and how that might aid in their formation. We're also considering doing looking at pre and post air expressing MTECs, so not just the MTECs that are in the air expressing stage of their development, and then also potentially perturbing the system, so altering the way that T cells are interacting with the MTECs in the thymus and seeing how that alters the way that air is localizing in the nucleus and what DNA sequences it's interacting with. So I want to acknowledge the Anderson and Gardner labs, which are very distinct labs. I'm sorry I put them together, both at UCSF for hosting me this summer and allowing me to perform this work. I did a lot of optimization on the SPRITE protocol since MTECs are very finicky cell type. And then the Chung lab, which is where I work at CU Boulder, for giving me all of the tools to work in the Anderson and Gardner labs this summer and for getting me here before you today. So thank you so much. I'm happy to take any questions now.
Thank you very much. Let's take one question, please.
Hi, that's really cool. I had a question about the Tmax cells and how the proteins actually get displayed to the T cells. Is it through HLA, or is it a transmembrane protein that actually displays these different cell types?
On the MTECs? What's expressing the TRAs?
The MTECs.
Yeah, so MTECs specifically express MHC class 1 and 2, and that's how they're presenting the TRAs.
Got it. Thank you.
Yeah.
Thank you so much, Kayla. Now we would like to welcome Margaret Orr from the University of Rochester. Welcome, Margaret.
Thank you, Carol, for that introduction. Hello, my name is Margaret Orr, and I'm a student researcher in the Gamma Gami Lab in the Department of Biology at the University of Rochester. and today I will be discussing N-terminal modification of methionine as a mechanism to combat oxidative damage. To begin, pathological aging and neurodegeneration are major health issues for humans and will only continue to become more prevalent over time. Here we see that it is projected that 13.8 million people will have Alzheimer's dementia, a type of neurodegenerative disorder, in 2060. And shown in purple, the majority of these individuals are projected to be over the age of 85. Thus, determining mechanisms to slow the onset of neurodegeneration and to slow aging will have great impacts for the human population. Proteins are the functional molecules of the cell, and they are composed of amino acids, some of which are susceptible to oxidation by oxygen radicals generated during cellular respiration, called reactive oxygen species, or ROS. And once these amino acids become oxidized, protein misfolding and aggregation can occur. And this is a hallmark of both neurodegeneration and aging. Specifically, methionines within proteins are highly susceptible to oxidation by ROS due to the sulfur in its side chain. And when methionine becomes oxidized, it is turned into methionine sulfoxide, seen in red here. And this is a form of chemical damage because methionine is a nonpolar amino acid, which is now converted into a polar amino acid, and this is changing the other residues it interacts with, causing this protein misfolding and aggregation described previously. Luckily, this chemical damage can be reversed by methionine sulfoxide reductases, or MSRs. And these repair enzymes will convert methionine sulfoxide back to methionine. And we searched for mechanisms to enhance this reduction and thus decrease the frequency of oxidative damage within cells. Specifically, we looked at modifications unique to methionine. And here we looked at formal methionine, shown in blue here. And formylation is present in bacterial and human mitochondrial protein synthesis. So the initiator methionine always contains this formal group, but it is not known why. And here we propose that this formal group's function is to prevent the accumulation of oxidative damage, and that when methionine becomes oxidized during translation, it is rapidly reduced by MSRs to prevent peptide misfolding and thus prevent protein aggregation. So to test this, we used mass spectrometry-based proteomics and E. coli as our model organism. So we extracted proteins from E. coli and digested them into peptides so that we had a mixture of formylated methionine and unformylated methionine. We then oxidized them with hydrogen peroxide to mimic the oxidation that occurs within cells, and subsequently reduced them with MSRs so that we had a mixture of formylated and unformylated methionine as well as oxidized and unoxidized. And then we analyzed this on a proteomic-wide scale by liquid chromatography mass spectrometry. And we found that formal methionine sulfoxide was a much better substrate for these MSRs compared to methionine without this formal group. And this is seen by the large decrease in fraction oxidized for the blue box plot. And here we determined that in vitro, formylation may be a mechanism to help cells combat oxidative damage and thus lessen the frequency of peptide misfolding and aggregation linked in Alzheimer's and neurodegeneration. So after this in vitro result, we now looked at in vivo, specifically in E. coli. So methionine tRNA formal transferase is the enzyme that adds this formal group onto methionine. And we knocked out the gene called FMT in E. coli that codes for this protein. So now E. coli was unable to formulate methionine, and thus formal methionine did not exist in these mutant cells. And we found that under normal conditions, these mutant cells were more susceptible to oxidative stress, as seen in the increase in PSMs, or oxidized peptides. And this phenomenon only intensified when we incubated these cells with hydrogen peroxide. So we see a large increase in the number of oxidized peptides And this is still in the presence of these MSRs, so there is a better reduction in the wild type cells. And after we saw that formylation and N-terminal covalent modification on methionine helped facilitate the reduction of oxidized methionine, we now look to other modifications, specifically acetylation, which is a common modification in human proteins. So here, this acetyl group is shown in green here, quite similar to the formal group, but just a little bit larger. And we completed a similar experimental procedure and received similar results, where acetylmethionine was much better reduced compared to methionine with no modification. So in conclusion, we know that proteins are the functional molecules of the cell, and they are susceptible to oxidation by reactive oxygen species. But MSRs can reduce this oxidation and prevent the accumulation of oxidized proteins and thus protein aggregation. And these methionines can also be formylated, shown in blue here, or acetylated. And when these methionines become oxidized, they are better reduced by these MSRs, which helps decrease the amount of oxidized methionine in the cell. And in the future, we plan to investigate the active sites of these MSRs and determine why these covalent modifications enhance their efficiency. Also, we can determine mechanisms to promote the addition and retention of the formal and acetyl groups onto the N-terminus of methionine for possible therapeutics, and thus help decrease the amount of oxidized proteins within the cells, and hopefully one day decrease the prevalence of neurodegeneration and slow aging. I would like to thank the Astronaut Scholarship Foundation for inviting me to speak today, as well as for their support, and the Gamma Gami Lab at the University of Rochester and the Mass Spectrometry Resource Lab for help with running my samples. And I'd like to take any questions.
Thank you, Margaret. Who has one question for Margaret?
I'm curious if you have any interest in studying, if I'm remembering my biochemistry correctly, cysteine residues, since they also contain sulfur groups.
Yes, thank you. That's a great question. Specifically, why we have focused on methionine is because cysteine oxidation is reversible without the use of enzymes. So in contrast to methionine, methionine needs to be reduced by MSRs, and different organisms contain different types of MSRs, which reduce different diastereomers. But we are looking for ways to enhance the reduction of methionine oxidation because it's not reversible without the use of enzymes.
Thank you so much.
Thank you.
All right, our next presenter is Kennedy Lynch from the Louisiana State University. Welcome, Kennedy.
Okay, full disclosure, the transitions don't work, so sorry about that. But hi, today I'm going to be talking to you about the development of diagnostic biomarkers for cerebral malaria, specifically in avian models. That's what my neuroendocrinology and behavior lab works with. And specifically, we're looking at gene expression of pro-inflammatory cytokines, as well as the ratio of angiopoietin-1 to angiopoietin-2, which I'll just call the ANG ratio from now on. So to give you just a quick and dirty introduction, avian malaria is a little known but actually very extreme issue that's affecting bird populations around the world right now. It's leading to a decrease in both biodiversity and population numbers overall, and it's only going to keep increasing as the vectors of climate change increase their ranges, so mosquitoes, biting midges, flies, stuff like that. And cerebral malaria is the most severe form of malaria. To give you a scope of how bad it is, it makes up about a third of all human malaria cases, and if it goes untreated, it has a 100% mortality rate. Avian cerebral malaria pathology is grossly understudied, and that's because it's really only been studied in humans, and mammalian and bird brains are extremely different in terms of like gray and white matter and neuron density and stuff like that. And so this leaves a really large gap in scientists' knowledge about it. And so to give you a little overview, there are 3 hallmarks of cerebral malaria, and as you can see, they're up here, and these are all interrelated and inform one another. The first that I'll talk about is parasite sequestration, and this is when the actual parasites invade the microvasculature of the brain, and that causes congestion of these vessels, and that correlates with cerebral malaria severity. Next, we'll talk about endothelial dysfunction. Because of this sequestration and the blockages in the microvasculature, the blood vessel integrity starts to degrade, and that can lead to the breakdown of the blood-brain barrier, which is not good. And the third is inflammation, and this is regulated by pro-inflammatory cytokines like TNF-alpha and IFN-gamma, and in previous studies in the lab, we have shown that those are positively correlated with malaria severity. And so our focus is going to be undertaking the first ever investigation of cerebral malaria pathology in birds. And so we're going to be, as I said, creating diagnostic biomarkers and using qPCR probes and primers to be able to diagnose cerebral malaria without lethal sampling, as that's the only way to diagnose it in birds right now is like taking the brain tissue and looking at it under the microscope. And so first, we'll talk about the angiopoietin ratio. This is an indicator of endothelial dysfunction. Angiopoietins are just endothelial growth factors, if you weren't aware. And so this Ang ratio is indicative of membrane permeability. Ang1 stabilizes endothelial tissue, while Ang2 does the opposite and increases the permeability of the blood vessels. So basically, we're using that as an indicator of like the breakdown of the blood vessels. And so why are we looking at the Ang ratio specifically? This has been shown to be associated with cerebral malaria in humans and can predict the severity of malaria. And it's also available to be detected in the blood, which is something that won't require lethal sampling, like I said. And then in terms of inflammation, we're going to be looking at pro-inflammatory cytokines. Cytokines are just signaling protein molecules that are involved with regulatory processes. So like they're pro-inflammatory cytokines, anti-inflammatory cytokines. Some anti-inflammatory ones are interleukins. You've probably heard those, IL-1, IL-4. And so, like I said, we're going to be looking at TNF-alpha and IFN-gamma. And in previous studies in the lab, TNF-alpha was positively correlated with parasite load. So as you probably know, parasite load is a good indication of how sick you are. If you have more parasites, you're going to have more symptoms. You're more sick. And previous studies also show that IFN-gamma was not correlated with parasite load. So we just have both sides of the coin there. And as I said, it's correlated with malaria severity, and this is also detectable in the blood, which is why this is also a good candidate. And so basically we have 3 different experimental groups: the infected birds, the resistant birds, which were exposed to malaria, and the sham donors, which were never exposed to malaria in the first place. And so just quickly going through the methods, I did section the brain tissue. We got some thin slices and thick slices. The thin slices, we were able to identify regions of interest. This picture is a picture of the cerebellum, which is what I'm going to be focusing on. We took samples, using a core to get RNA. And so right now I'm about to start the RNA extraction process. And so then we're going to use that RNA to do qPCR and target the antigens and as well as TNF-alpha and IFN-gamma. And then we're just going to compare the results of gene expression between the different experimental groups, as well as correlating it to circulating parasitemia from blood smears that we took in the field. And so why is this important in the first place? There's 3 main reasons. 1, this method of creating biomarkers can be used in human health. 2, we need to conserve avian biodiversity. We're the stewards of the environment. And 3, this will actually help us be able to get an accurate figure of how many birds are suffering from avian malaria. And so, yeah, as I said, this is an ongoing project, so I'm not finished, but I will be completing it very soon. So thank you, and I'll take questions now.
Thank you. Who has a question for Kennedy?
Hi, I was wondering, so in your study you only use house sparrows as a study species, but you're looking to measure like levels of other birds, I'm assuming native birds to the US too. Do you have any plans to expand to some of the other species? Like how are you planning on, I guess, inferring that based on just one?
Yeah, so house sparrows is like a model that we use a lot. We have to use species that we can like have readily available to us. They catch these themselves, like they go out in the field and catch them. We do a lot of work with European starlings as well, but this is just the first phase of a long series of projects. as I said, this is the first investigation into cerebral malaria pathology in avian models, so we would like to expand later down the line, but this is just a first step.
One more question.
So you mentioned, I think you're taking samples out of the cerebellum.
Yes.
Why did you choose that part of the brain as opposed to any other?
Yeah, so we originally had like a bigger scope for this project, but this is going to be my senior thesis, and I'm like, I do not have time to do that. So we just kind of chose one. The cerebellum is like a big area. It's easy to identify. One thing that I wanted to target was like the hypothalamus, but that was like what I wanted to do specifically because malaria does cause like extreme like sleep-wake cycle like interruptions, but there's like one very specific neuron that we were gonna have to locate, and it was gonna be really hard to like get that out of the brain tissue. So the cerebellum is one that's easily identifiable with staining. It's a pretty big area, so it's very easily it's easy to manually punch. So that's just the first thing that we're gonna do. We eventually wanna do the hippocampus and the striatum as well.
Cool, thank you.
Thank you.
Thank you, Kennedy. All right, let's welcome Noelle Naughton from Purdue University. Welcome, Noelle.
Probably stand in front of the mic. That'd be a great idea. So hi, I'm Noelle. I'm a senior at Purdue University studying biochemistry. And this past summer, I was at the Indiana University School of Medicine under the mentorship of Dr. Steve Angus, studying how FAC is a potential treatment of malignant peripheral nerve sheath tumor. And so in the Angus lab, we study neurofibromatosis type 1, or NF1, and this is a cancer predisposition syndrome that affects roughly 1 in 3,000 individuals. And so the NF1 encodes a tumor suppressor gene called neurofibromin, and this is a negative regulator of the RAS family of proteins, which are known to regulate cell growth and survival. And so whenever you have like functional loss of neurofibromin, this leads to the amplification of RAS signaling, and this can lead to the formation of these benign tumors that can form in the peripheral nerve system. And so these benign tumors are called plexiform neurofibromas, or PNFs, and this occurs in about 50% of NF1 patients. And in these NF1 patients that develop these these PNFs, about 10% of them undergo a malignant transformation to a rather aggressive and refractory type of sarcoma referred to as an MPNST. There we go. And so many patients experience different manifestations of this disease. However, we are most interested in malignant peripheral nerve sheath tumors, or MPNSTs, because they are the leading cause of premature death in NF1 patients, and currently there are no approved targeted therapies to treat this. So it's of high interest to develop effective therapies for these patients. And so to kind of approach this, I turned to some past studies and identified a drug called cabozantinib, which proved to be effective in treating NF1 PNFs, and so this kind of normalized nerve microarchitecture as well as reducing tumor volume. And so some other groups found that there's increased phosphorylation in FAC, which stands for focal adhesion kinase, and this is known to regulate. It's a protein that kind of, I guess it's for signaling it promotes a signaling cascade related to cell growth. And this was found to be increased in response to cabozantinib treatment in hepatocellular carcinoma. And then another group found that if you inhibit FAC and you also have cabozantinib treatment, this leads to a demonstrated therapeutic synergistic effect. And so with these pieces of data, this really generated some enthusiasm in the lab to evaluate not only cabozantinib, but also targeting FAC to be able to develop new therapeutic avenues for NF1-associated malignancies. And so first what we wanted to do is we wanted to assess FAC expression in different tissues for the progression of like wild-type tissue. There we go. Wild-type tissue to neurofibromas to MPNST. And so the yellow shading that you're seeing is the amount of FAC abundance in these tissues. And you can see that the MPNST tissues have way more yellow in comparison to the other two tissues. And so based on this finding and our interest in combinatorial studies, we turned towards a drug database inquiry. And so the first drug that I talked about, cabozantinib, it shares many overlapping drug targets with another drug called crizotinib. And this is FDA approved for lung cancer. And the notable thing about this slide is that both these drugs target PTK2, which is the gene name for FAC. And so this got us really interested in evaluating the efficacy of crizotinib in treating MPNST cells. And so what we did is we did some cell viability assays as well as some colony forming assays. And we can see that with crizotinib treatment, this seems to wipe out, colony formation. And so this is a really exciting fact or finding, because this could have, clinical translations into the clinic, and this could be a new way to treat NF1-associated malignancies. And so to, confirm that if crizotinib truly does inhibit FAC, we turned towards kinome profiling. And so on the left-hand panel, these are kinases that show decreased binding. And so the way that we can interpret this is that crizotinib is directly binding the FAC and thereby preventing it from interacting with the beads, since this is through affinity chromatography. And so we also, similar to the crizotinib finding and it impairing human MCST cells, we also wanted to evaluate if FAC inhibition would elicit a similar response. And so through two methods, pharmacological inhibition and siRNA-mediated inhibition, we can see that there's a reduction in colony formation as well as luminescence with another assay that we did. And so this shows that both these two methods are effective in impairing human MPNST cell growth. And so to kind of circle back to our initial interest in combinatorial studies, we evaluated the combination of defactinib and cabozantinib, and we can see we did some synergy screens. And with 4 independent measures of synergy, we can see that all of these scores have a score higher than 10, and that fits the criteria of demonstrating a therapeutic synergistic effect. So this means that these drugs have a multiplicative effect on each other and enhance each other's effects rather than acting, independently of each other. And to also kind of give a visual guide of this, we did some colony-forming assays in 2 different MPNST cell lines, and this shows that as we progress from untreated to the single agents to the combination, we can see that there's a decrease in colony formation. So that can kind of give us another visual cue of synergy. And so in the future, we also want to conduct some in vivo studies with defactinib and cabozantinib. And if this internship were longer, I want to do some more preliminary in vitro studies with crizotinib and the other FAC inhibitor, VS4718, and try to, hopefully all of this data in our future work will help us with identifying new attractive molecular targets and developing effective and novel combination therapies for NF1 patients. And so I just want to acknowledge the Astronaut Scholarship Foundation for allowing me to be here, and the 3 highly collaborative and adjacent labs that helped me and mentored me at my time at IU. And I am happy to answer, I'm happy to answer any questions.
Thank you so much, Noelle. We have some questions.
Hi, Noelle. Great presentation. I was wondering if you could expand a little bit on what you meant by MIB binding about halfway through.
Yeah, so I did not have time to explain that in the method. So MIB stands for multi-inhibitor bead binding coupled to mass spectrometry, and that's just a really fancy way of saying that we can capture kinases and their active conformation. And so what you do is you can take cell lysates, you could take patient tumors or mouse tumors, and you could run them through a column that have these beads that are coupled to an array of different kinase inhibitors. And this allows us to kind of maximize the global kinome and collect all the active kinases, and then mass spectrometry is a way for us to identify and quantify those kinases.
One more question.
Thank you for your talk. I also have a question about this volcano plot here. So it looks like you do have off-site targets as well, which you talked about earlier in your presentation. But were these the ones that you expected? Is first part of my question. And second part is, do you think that this will still be a viable drug even though you had other genes upregulated and downregulated?
Yes. So not necessarily always, because, for example, crizotinib is FDA approved for lung cancer, and it's known to historically be an ALK inhibitor. And so as you can see, ALK is not upregulated on here, it's not downregulated on here. And so that might be really surprising. And so a lot of these kinase inhibitors are type 1 inhibitors, so they competitively compete with ATP. As kinases, their active site architecture and sequence are highly conserved. And so they only bind to ATP, so that's why you see a lot of those off targets, and that's like an ongoing issue with type 1 inhibitors. However, there are type 3 inhibitors that are allosteric inhibitors, and so those bind to a site that might be more unique outside of the active site. And so that's one avenue that people are looking at to make these kinase inhibitors more targeted and specific. So.
Great. Thank you so much, Noelle. So our next presenter that we're going to welcome is Manas Gupta from the University of Texas at Austin. Welcome, Manas.
That's not my presentation.
We have an AV issue. So this is Brandon's presentation. I think we might need to shift to the correct PowerPoint, so please stand by.
Should I just keep clicking all the way through? No. Do I just keep going all the way through? Yeah, okay.
Sure.
This may take a minute.
Yay. One moment, please. Processing.
This is that the one you gave me?
Yeah.
I don't know why it did not do that, but you can just roll with it.
Thank you to AV for everything they're doing, and there you go. We're ready for Manas.
So today I will be talking about how synthetic cells with connexin 43 can synchronize stem cell-derived cardiomyocytes. So a little bit of an introduction. Heart disease is one of the most prominent causes of disease worldwide. We have over 600,000 cases in the US alone yearly, and around 17 billion across the world yearly as well. In one specific case, myocardial infarction or heart attacks, over 1 billion adult cardiomyocytes die immediately and are replaced with non-contractile scar tissue or fibrotic tissue. And the cardiomyocyte population is unable to regenerate endogenously and requires potential exogenous cell stimulation. Cardiomyocytes derived from stem cells, specifically iPSCs, provide a high amount of potential to restore this host tissue. However, iPSCs are immature functionally, genetically, and structurally, and have, on the functional level, have extremely low electrochemical coupling. Specifically, when you implant them into a mouse, a human, they will cause really, really bad dysrhythmias that are fatal. So what is electrochemical coupling? Electrochemical coupling is the fact that adult cardiomyocytes have a wide amount of gap junctions, or specific junctions that connect cardiomyocytes, allowing for communication between the two cells and for them to contract at the exact same time. Once again, iPSCs do not have that ability, especially the ones that are immature and don't have any sort of functionality on that spectrum. And thus, if you transplant functional tissue that is made from these iPSC-derived cardiomyocytes, they will result in dysrhythmias causing over 120 beats per minute, leading to fatality as well. So we hypothesized that by increasing the amount of gap junctions within the cells, by using a synthetic delivery system, we would be able to allow for better utilization of these iPSCs for heart attack patients. So our approach is connectosomes, which is what we dubbed our synthetic cell delivery of connexin 43, which is the main gap junction found within the heart. So if we look at the figure above over here, we have a synthetic cell, and all the green on the outside are going to be your gap junctions. And these are harvested from any sort of stem cell, specifically iPSCs. And by having the high expression of connexin 43, if we were to dose them to adult cardiomyocytes, we are expecting to see that there would be increased retention of these gap junctions and formation of actual syncytiums, as seen in the bottom right corner, that connect the actual cardiomyocytes and allow for electrochemical coupling. So in order to actually generate these, we went through a large generation process as well as standardization and things like that. So the actual generation process, if you look in the bottom corner over here, the white arrows very directly show you where the connectosomes are actually made. And then in terms of actually characterizing it, we looked at various different types of cell types and showed that the connectosomes themselves have the same exact size amongst all different cell types that connectosomes are derived from. Further, overexpressing your connexin 43 will not actually discredit the size as well.
Further, cytotoxicity. If we're dosing to the cells, we don't want them to actually die as well. And dosing our connectosomes causes for no cell death across the board as well. And finally, we want to make sure that our connectosomes are actually being delivered to our actual cardiac tissue. And by doing in vitro experiments on these, we're able to see co-localization of the green for our connexin 43, as well as orange that is actually inside our vesicle to show that all of them are inside the actual cardiac tissue, seeing that they're actually being delivered to the cells. So from here, we're going to look at a little bit of functional analysis. And here, this slide looks directly at no connectosomes at all, and we're looking at synchronicity as a measurement here. If you look on the figures to the right, you see actual cardiac tissue that is beating, and the green that is beating is as showing a calcium ion being bound and fluorescing as well. When you have connectosomes, you see significantly increased synchronicity as you go down to day 8, and that's kind of quantified on the figure over here as well. Moving on to region of interest studies, now we're actually able to look directly at specific cardiomyocyte populations and see how whether distinct populations increase in synchronicity after dosing. And as you see, amongst all of them, we see that connectosomes cause for long-term synchronicity increases. Last but not least, we're looking at the future studies and the key takeaways. And the first main key takeaway is that our connectosomes do allow for synchronicity increases. And the second is that these synchronicity increases are long-lasting, so they occur well past the 8th day. So for future studies, we are looking at investigating a freeze-dry mechanism that allows us to store these for long-term usage. And further, we are going to be studying these in mouse models using a coronary artery ligation model in the future as well. And I'd like to acknowledge the Zolden Lab, especially my mentor, Dr. Janet Zolden, and the postdoc, Dr. Nima Mahatman, for their mentorship as well. And I'll take any questions at this point.
Thank you very much. Who has a question?
So this might be a basic question. Sorry if you mentioned it in your presentation, but what happens if you directly transfect the iPSCs with this connexin 43? Do you need synthetic cells to bring this protein payload?
If you directly transfect them into the iPSC, we find that there is very high recycling rate of the connexin 43. So the gap junction will go to the actual membrane and be recycled pretty immediately and not having that sort of long-lasting effect. Whereas if we have that synthetic cell delivery mechanism, we see that the post-day 8 mechanism of long-lasting synchronicity benefits as well.
Gotcha. Thank you.
Any other questions? One more.
A really interesting talk. I'm excited to hear about how this therapeutically could be dosed. Would it be surgically implanted, or how would this technology be used?
Yeah, so in the rat models that we're going to be using, actually this upcoming semester, we're going to be looking at direct injections into the heart, where the actual myocardial infarction that we're inducing is going to be looked at. So that's currently what we're looking at, but as I actually go through in vivo studies, that might very much change.
Thank you.
All right. Thank you so much, Manus. Very interesting. Next, we would love to welcome Chris Barron from Texas A&M University. Is it Longhorns? No, sorry. Oh, don't boo me. I don't know. Aggies. There we go. Aggies.
Thank you. Yes. Howdy, y'all. My name's Chris Barron. I'm a senior at Texas A&M University, double majoring in plant and environmental soil science with a crops emphasis and biochemistry. So the scope of this talk is going to be very different from everything else that we've seen. But so basically what I'm working on in my studies is to develop a protocol to assess drought tolerance in cotton. So first of all, why do we care about this? Well, we grow a lot of cotton in Texas, and we also grow a lot of cotton in the USA. So the US is currently the 4th largest producer of cotton globally, and a lot of that does get exported. Also, cotton is a globally important commodity. Many of you are wearing it today, and so fiber is one of the most important industries in the world. Texas produces a quarter of US cotton, and much of the cotton that's grown in Texas, US, and in the world is grown in areas that are prone to drought. So this is a drought map of Texas last year in August. And the dark red areas here and here are actually the highest production areas of cotton in Texas. You'll also notice that those overlay the Ogallala, and also we have the Trinity Aquifer over here. This is a high production area, not cotton, but other crops. And so both of those regions rely heavily on those aquifers for irrigation, and those aquifers are very quickly being depleted. And so basically, we need to figure out how can we still keep producing cotton, but not depleting our natural resources. And so one of those ways is to develop a more drought-tolerant crop. So the goals of this research are first to develop an effective and fast protocol. Traditionally, with normal conventional drought tolerance breeding programs, you need at least 10 years before you can assess if a crop is truly drought tolerant. We want to discover physiological characteristics specifically in cotton seedlings that can be correlated to stress tolerance. And then most importantly, we want to figure out how we can quantify those. Drought stress and drought stress tolerance are not as easily quantifiable as, say, yield or even nutrition content. And so a big focus of this study is to quantify drought stress. And then finally, the most important part is I want to correlate drought stress response in a cotton seedling to drought stress tolerance in a mature plant. And so the outline of this experiment is first there's going to be a baseline study that kind of establishes parameters. So that's what I'm working on right now. And so our drought tolerant check is a Phytogen 400. So that's a long season variety known to be drought tolerant historically. And then we also have our susceptible check, Delta Pine 2115. That's a shorter season crop, tends to need to be on irrigated land, and so it's more susceptible to drought. For the test groups, there are 10 groups of 5 plants for each genotype. 5 of those groups are going to receive a drought stress treatment, and the other 5 groups are going to be a control group. And so basically, it's going to be comparing the response between those groups and kind of establishing this one had a much more severe response, this one had a much more mild response to the drought stress. And the stress markers that I'm specifically looking at are going to be chlorophyll, leaf temperature, which is related to wilting, and then also stunting. And so the plants are started on their drought stress at V5, so that's vegetative growth stage 5. At that point, a cotton plant is reproductively mature, and so the response should be similar to a mature plant. And then the stress is applied over a 2-week period, followed by a 3-day recovery period. So currently, I'm running the first of 3 replications to establish the baseline trial. This is going to be followed by a follow-through study, or kind of a proof of concept, that is going to be using 4 RIL, so recombinant inbred lines, 2 of which are going to be drought tolerant, 2 of which will be susceptible, and the goal will be to see, can this protocol re-identify which of those lines were tolerant and susceptible. And so after completing the 3 applications, I'm going to identify which of the traits I'm measuring most clearly indicate tolerance or susceptibility, and then use those for a more streamlined protocol to assess those 4 lines. And so this is actually from day 7 of the current replication that I'm on right now. And so these are both our susceptible check, but this is the control group, and then this is the drought stress treatment group. And so what we can see here right now, these pictures are used to assess wilting, and so there's quite a severe response, are only halfway through the drought stress period. So hopefully this is a good indication that this will be able to identify it. And so just some thank you to the Dr. Stelly and Dr. Vaughn are the leaders in my research lab, and then also Dr. Haig, Dr. Smith, and Dr. McPherson and Dr. Hopkins are cotton breeders who have given me some insight into what parameters I should be testing. And then also Mr. Scott Adair, I've worked with him in the Corteva's corn drought tolerance testing program in the past, and so he's given me some really helpful insight as well. All right.
Thank you so much. Questions?
Have you considered any factors that might be related to like the roots or less visible on the surface?
So unfortunately, root mass is a very good indicator of drought stress tolerance. And it's not something that I'm going to take into account into this experiment, because basically the goal of this is that it can be easily done on a large scale. So it should be easy to run this protocol with, say, 100 genotypes, not just 4. And unfortunately, analyzing root biomass is very difficult. It also limits the amount of substrates that you can grow in. So generally, if you want to analyze root biomass, you need to grow in sand. Unfortunately, sand dries out very quickly. And so by using a soil substrate in this, it dries out over the 2-week period, and so there's no need to be constantly rewatering the plants, and so it allows a more accurate representation of a drought stress that you would get in the field.
Awesome.
Any other questions?
Yeah. Is it just coincidence that cotton happens to be growing in drought-prone areas, or is there something particularly resilient about the cotton crop to droughts?
So cotton is actually a perennial crop, so it will grow indefinitely, but we grow it as an annual commercially. It's just more efficient that way. And so cotton itself actually is already quite drought tolerant, and that's why there's been very little research into developing drought tolerant lines already, and so there's been also very little optimization in the drought tolerance breeding. So basically, this is the development of this protocol is kind of anticipating a resurgence now in developing drought tolerant lines, and so the goal of this would be if you have a lot of new novel genotypes, maybe with some introgressions from wild DNA, to quickly screen a large variety of those and determine, okay, these have potential for drought stress, these don't, and then move those on to a more rigorous testing program. All right.
Thank you so much. And now we are going to welcome our last presenter for today, Christopher Tomo from Tufts University. Welcome, Christopher.
Hello, everyone. So I'm going to be talking about identifying chemical weapons that fungi use to spoil our food. It's kind of a flashy title, but I basically take a multidisciplinary biological approach to understanding poorly characterized organisms in food. Let me skip this slide. Let's talk about cheese. So we've seen cheese in the grocery store a lot. We see these wheels with the pretty white rinds. This is a camembert. On this cheese curd biofilm, there's a stable community of about a dozen microbes that kind of peacefully coexist on the rind. So it's bacteria, fungi, yeasts, all kind of living together in harmony. I studied these for about a year and a half, and then I got bored, and I said, let me do something a little more chaotic. So I study food spoilage. I don't care about this stuff. I don't know what happens when you throw in this really crazy, really toxic fungus, and that just messes everything up. So that would be A. West, Aspergillus westerdyki. I just use A. West because it's shorter and less complicated to pronounce. It was discovered in 2004. It is not really well characterized, especially in food systems. All that it's really been studied in is in your house and in like your little vacuum cleaner thing that you don't clean out. It's kind of known for causing like respiratory illness. So we know that it does produce very potent toxins. We just don't know how this actually plays out in food systems, let alone on the cheese rind. So it's been isolated from a bunch of different food products, aged meats. You can see this very pretty picture right before we eat of this aging meat with the yellow fungus growing all over it. Cheese, which we're about to talk about, coffee beans, and then as well as crops. This is also very commonly found in plants and soil. So this is what a cheese rind will look like in an aging facility, which is a little bit different than we're used to seeing. We kind of see that pretty grocery store ready cheese rind that we saw before. This is kind of what it looks like in an aging facility, so quite gross, lots of different patches of growth, but there's still kind of like a uniformity to it. When this cheese rind is contaminated with A. West, it looks even grosser. There is this black growth all over the cheese rind. It is clearly wiping everything out. In the bottom right, we see this zone where absolutely nothing is growing. There is no native cheese rind. And obviously, this would not be put out to consumers, but for cheese producers, that is a loss of money, a loss of profit, and also we don't really understand why this happens. So my research is kind of taking a multidisciplinary approach to understand how do we get from this rind that is ready for sale to something like this? What is actually happening in that rind community to affect the cheese? So I'm taking 3 approaches, the first being genomics. So what do genomic and transcriptomic data reveal about how A. West lives on cheese and how it impacts the native microbiome? A genetic and mechanistic approach, so what genetic mechanisms are underlying these observed antimicrobial activities and how can we manipulate the genome to study these phenomena? Filamentous fungi are notoriously hard to work with in genetics. When I did bacteria and stuff, I could be doing transformations in about 1 or 2 days. Now it takes like 3 weeks and usually doesn't work half the time. So I'm trying to figure out how to work with the genome. And then lastly, I do ecological experiments. So how do the different microbes native to the cheese rind respond to AOS contamination? So I do that with pairwise experiments between 2 microbes as well as with the entire community as a whole. So I'm mostly going to be focusing on the genomics for today. So I use an RNA sequencing data set, which is where we grow this fungus in media in 3 different conditions. So the first being AWest alone on malt extract agar, which is a plant-like media that kind of simulates a more general environment. Condition 2 being AWest alone on cheese curd agar, which is our sort of in-house way to simulate the cheese rind because we can't actually just have giant wheels of cheese in our lab. And then the third condition is AWest on cheese curd agar with a simplified microbial community of about 4 species. So we grow these up for about a week. We collect all the RNA from it. We send it off for sequencing. And then I just do a bunch of bioinformatic analysis. So the first comparison I'm going to be making is between conditions 1 and 2, which is AWest alone on this plant-based media and AWest alone on this cheese media. So the question that I'm really trying to get at is, how does AWest respond to living on cheese versus a more general environment? This is a volcano plot of differential gene expression. So every single dot on this volcano plot is a gene, and everything that is in orange is significantly differentially expressed. So about 33.6% of the genome is significantly differentially expressed, which is insanely high for microbiology. It is usually not this high at all. So we know that cheese is kind of a stressful environment. It has to really adapt a lot and change its sort of natural processes. So I kind of wanted to dig deeper because that's still a mess. And when you're working with a non-model organism like AWest, there isn't a lot of great information out there. It's a little bit easier if you're doing something like yeast or E. coli, and the genome is well annotated. But for this, there's basically nothing. So I used a software called Cobos, which kind of takes the differential expression data and maps it to known metabolic pathways. I'm not going to get into all the specifics of it, but the ones that were really significant were differential expression of nutritional pathways, so especially amino acid metabolism and lipid metabolism. This kind of makes sense for living on cheese because the amino acids in cheese aren't freely available, so AWest has to work a lot harder to kind of produce its own amino acids because in soil there's a lot of nutrients readily available. Cheese is growing in a highly saline environment. In a cheese cave, not a lot of things going on there. And then the other thing that I noticed, which is very interesting, is differential expression of secondary metabolic pathways. So secondary metabolites are chemicals produced by mainly fungi, and they aren't required for development or maintenance of normal functions. So these are considered like specialized chemicals that are for extraneous processes, for example, antimicrobials. So I dug a little bit deeper into these, and I'm not going to be talking about this research today, but I'm happy to talk about it after. But we identified a couple of different antimicrobial products that were directly linked to this expression analysis. The second comparison that I made was between conditions 2 and 3, which was AWest alone on the cheese curd agar, and then with the simplified microbial community. So this volcano plot looks a little different. Basically, nothing changes. And that is a little interesting because we saw this huge difference before, and my main takeaway from this is that this fungus is so powerful, it really does not care what is going on with the other microbes. You know, those are the ones that are affected. This fungus is just chilling. It is doing its thing as normal. It has it really doesn't recognize that there's other microbes there. It just sees something else, kills it. So I imagine that if I were to do further transcriptomics with the bacteria from this data set, those things would be significantly upregulated, and those volcano plots would look a lot more like the first one. So my second approach is genetics. So I am trying to figure out how to manipulate this genome and sort of make the connection between this antimicrobial activity and, is there a specific gene or set of genes that are being differentially expressed when it's in the presence of other microbes on cheese. So I'm currently working on optimizing a gene replacement protocol for transformation. So I have this target gene that I just found in the literature, LAA. It is a pretty well-characterized fungal global regulator of secondary metabolism. So it's known for producing a lot of antibiotic compounds in a bunch of different fungi. It just has not been studied in AWest. And in my bioinformatic analysis, this gene was significantly upregulated in that first data set. So I use a synthesized DNA fragment that is about 4,000 base pairs, which takes like 9 PCRs, and it's awful every time. And it has the selection marker that is kind of similar to the uracil synthesis pathway in yeast, so I can grow it in differential media and hopefully transform the fungus. I haven't been able to get this successfully going. That was kind of what I spent like the last month doing. But once I'm able to successfully do this transformation, it'll open up a whole bunch of new avenues to studying the effects of LAA deletion and other secondary metabolites. And then the last lens that I take is ecological. The last lens that I take is ecological. I'm not really going in depth on these, but I do a lot of pairwise experiments, growing things in cold culture. I have little tubes that have little tiny cheese rinds in them, and I grow them together and I kind of see what happens. I also have these other experiments where I grow this fungus on a plug of cheese. I put that on a lawn of responding bacterium so that only the metabolites can diffuse, and I see like zones of inhibition. So future directions, I kind of mentioned before that I kind of want to study the transcriptomic responses of the bacteria to this fungus. Genetics, I'm still working on successfully knocking out that gene and kind of harnessing this protocol that doesn't really work in filamentous fungi yet. And then ecologically, I have a whole bunch of experiments that I'm working on. If there's one main takeaway, we don't really know a ton about spoilage molds, especially in cheese or in a lot of foods. We know how to prevent them, which is great, but we don't actually know a lot about what is going on in the cheese rind. In sourdough, I've also studied this, so there's a whole bunch of different systems that we really don't know a lot about food spoilage mechanisms. And then I just wanted to acknowledge all of my different mentors across this project, as well as the ASF, and then all of you guys for enduring this talk about spoiled food right before we eat dinner.
Thank you so much. Who's got a question?
Hi. So I don't know a lot about like this genus of like fungi or anything, but have any other that you know of like fungi in this family or genus been investigated for therapeutic use, for antimicrobial use? I know you said this one causes like lung damage, so not that one probably.
So Aspergillus fumigatus is probably the most well-known fungus from this genus. It hasn't really been studied a ton in therapeutics. I would say the most similar filamentous fungi is probably Penicillium, which we all know from penicillin. So I would say that the Penicillium family is a lot more well-studied for therapeutics. It's also just a lot easier to work with because we know a lot more about it. I imagine that you could do something like that, but I'm not sure that anything like that has been done yet. Yeah.
Thanks, Christopher. Super interesting talk. So can you, now that you have a second, can you tell us a little bit about the ecological, like pairwise comparison experiments, maybe some of the big takeaways you've seen?
Yeah, so I do these cold culture experiments where I grow A. west and 12 other fungi in a little cheese curd tube, and I grow them pairwise. I've seen a lot of pigment production when it's in the tubes where this fungus kind of takes over. So I'm also trying to determine if there's maybe a gene that is related to pigment production and antimicrobial production. So I'm trying to figure out kind of the link between those, but generally, like, it all matches what we see on the cheese. Like, anytime something goes up against this fungus, like, nothing can really put up a fight. The only ones that can kind of persist are a couple of yeast species and then a couple of other filamentous fungi. So Penicillium is also pretty antimicrobial, so those two can kind of coexist, but it's very volatile. And some of them, like Penicillium, totally wiped A. west out, and the other ones, it was vice versa. So it's still kind of in the early stages, yeah.
Neat, thank you.
Thank you so much, Christopher. Yeah. Who's hungry? I'm going to rethink the Swiss cheese I put on my burgers from now on. So we are now concluded for day one of our technical conference. You are all amazing. Thank you so much. Let's hear a round of applause for our scholars. So a couple of public service announcements, if you will. So first of all, for those of you who presented today, I am a great note taker. I have a couple of comments for all of you. If you're interested, come see me up here or in the bar. No, I mean, come see me up here. So for those of you who are presenting tomorrow, some of you I got a chance to spend time with today. For those of you who do want a little bit of extra coaching, I would be very happy to meet you at some point between now and tomorrow morning. I want to give you a couple of reminders. First of all, most of you, I would say 95%, stuck to the 5 minutes. A few went to 6, a couple went over. So just in your mind, as you're prepping tonight, think about 5 minutes, okay? up to 6. And then, of course, we have a couple of questions. We are spot on time, so thank you all for that. And let's continue that for tomorrow. Number 2, please be sure that you're acknowledging your people that are supporting you, and most of you did that. So just an FYI, make sure that you're thanking the people and your mentors and the labs, et cetera, which is because we want to hear who's behind you and who's helping you to do all the wonderful things that you're doing. There's a fun fact scavenger hunt that is due at the end of the conference tomorrow to either Lisa or Janelle. So thank you so much. I absolutely loved being your host today. Again, I'm here for your comments. I'm here to practice. And then we do have a reception this evening at 7. Yep. So thank you all very much. It was a wonderful afternoon.
So Carol covered everything, but let's give it up for our moderator, Carol and Ashley. Thank you so much. Kept us on time. Volunteers, you did a fantastic job running that microphone everywhere. Well done. Everyone did a great job but Mason. No food for you tonight. If you know, you know. All right, everybody. Thank you so much. Scholars, 7:30. All right? Thank you. Bye.